remember to use the microphone. I think I have a little echo. Thank you. The virtual attendees, in order to ask questions, please use the Q&A button and raise your hand if you have any questions or comments. We'll be checking those. And then I also want to thank Amelia, Megan, Summer, and Kristen for all of their hard work in putting this meeting together. If you have any issues or questions, please reach out to them. So we do have CME for this conference. I'm not going to go over all of the details, but for pharmacists, it's 16.5 contact hours, and for physicians, it's 16.5 credits as well. There are some disclosures to make. I won't go over them all because there are so many, but you can see them here. Everybody's doing a lot of great work with industry and within respective committees. And then as far as our meeting, it's over the course of 3 days. On Wednesday, today will be essentially about career development and communication skills for our early career investigators and attendees. I wasn't, last year that I was in the same position. So this is the time where we can all just learn with each other, ask questions, please, and be sure to, if you do have questions, there are plenty of people here that have the expertise to answer them. And so please reach out to those respective people. Tomorrow will be presentations by the MPRINT Hub Centers, and then Friday will be discussion sessions on a variety of different things, including embedding and enabling human milk research in the NICHD networks, best practices for pharmacokinetic studies and reporting to support reproducibility, data sharing consensus, and gaps and opportunities in the vision for MPRINT. We will have a number of networking opportunities, breakfast, lunch breaks, as well as a Thursday dinner. We do have table assignments for those, so please make sure that you're going to the right tables when you do go to them. As far as our agenda today, we have a full day of presentations. We'll be starting with our presentation with Victor Nizet, and then we will follow that with our flash presentations by our early career attendees, followed by a break, and then our breakout groups. The breakout rooms are on the side over here. If you do not have your room number, please find one of our people, Amelia, Megan, Kristen, or Summer, to figure out those rooms. We will at 11 a.m. have a presentation about media, social media, which is an important thing that we all have to consider nowadays, followed by lunch. And then we will have our keynote speaker about servant leadership and overcoming burnout, as well as a presentation on choosing the right journal for your paper. We will again have a break, and then we will have a career development panel discussion, followed by an optional UCSD labs and facilities tour, which I hope everyone takes advantage of. I know I will. So I'd like to present our first speaker, and this is Dr. Victor Nizet. Let me just pull up your bio really quick, Victor. Okay, so Victor Nizet is a physician scientist and a distinguished professor of pediatrics and pharmacy at the University of California, San Diego, where he also serves as the vice chair for basic science or for basic research in the Department of Pediatrics, and is the chief of the division of host microbe and systems and therapeutics. And so today he will be presenting on Presentation Skills 101. Thank you, Victor.
Here in San Diego. And I actually know that Sarah gave a talk online to the MPRINT community about how to give the perfect 10-minute presentation. And I thought, well, I really can't improve on that very much. So I wanted to focus really kind of on longer presentations that you might give, or the kind of presentations you will do for the trainees in the future, maybe giving a job talk for the professors when you're a visiting faculty member, or you may get to give a longer address at a university, or you're going to present to try to recruit trainees to your lab, those type of presentations. And this little Okay. So, and I will also regain time. Sorry about that for connecting directly. I should have maybe just one on the Zoom, but I'm going to talk about observations I've had over giving presentations over the last 25 years. This one really put into focus how important planning your presentation was, because I was speaking in China. This was in an audience of about 800 people. And not only were there 2 copies of my slides behind me, there was a live video of me, and then I'm looking at a live video of me and like a mini jumbotron of my presentation. So the level of self-consciousness was astronomical, and every single wordy slide and glitchy transition really hit me. And I made so many major revisions to my kind of pocket presentation after this talk. And I think a couple things that I just want to mention. There's a lot of, I used to, come up with a new slide look depending on my mood for every talk. But I really think that there's a lot of science actually in terms of the audience grasping and the visual acuity. And I'm now appreciating this as I had my 60th birthday this year, that using dark colors like black or dark blue on a white background is always clean and safe. It has high visibility. And some of the important aspects of this, it's really easy to import graphics into that setting. It's much nicer to have a white, like I could have clipped this directly as a screen capture from a paper, imported it into the white background, and it looks completely clean, that data or many stock images and clip art that you might find. Whereas having a blue background or a textured background and then a white border around what you've imported, I always think looks a little bit sloppy. But an alternative, and it's really up to preference, is white or yellow on a black background, which is also very safe and highly visible. And one of the advantages of this is there's science that says in very large auditoriums, this is especially effective. And if your data is going to have a lot of bright images that are important to you or stylistically an aspect of what you do, especially the fluorescence microscopy intense data, that's a key component. The resting the eyes with the black background tends to make that much more discernible and effective, as you see here. What you definitely want to avoid is anything where there's not enough contrast for you to see well. I've seen blue on green, red and blues that don't look very good, even though they might look good to you on your laptop up close when they project on a big screen. Anything with too many colors, I think, is a disaster. People think that, oh, I'm going to differentiate a bunch of features with all these different colors, but they're only going to be up for a short period of time, and it's information overload, and it looks sloppy. And I think never use a photo as a background, even if it's super topical, like, you know, the cells or neurons you're studying, and you gray it out. It just takes away from the presentation. And I would take that a step further and really think twice about putting an institutional logo on every single slide of your presentation. The university will often request this. Probably 30% of the presentations you see will have this, but it takes space, and it's really not adding to your presentation, especially a short presentation. You're going to give, you're going to declare where you're from and thank your institution at the end. I think people will have an understanding of that. And any kind of repetitive graphical element, DNA helix, I see about 5% of presentations in the field of genetics. It's kind of cool, but over and over again, honestly, it becomes distracting to the audience. And I think this graphic designer came up with it. Do you really need a logo on every slide? It's a presentation. You're not a NASCAR racer sponsoring a particular institution. There's research also to suggest that you should be using sans serif fonts. Those are fonts that don't have the little decorative ditzels on the end of V's and C's sometimes. They are easier to see than serif fonts like Times New Roman and other colorful fonts. So that is strongly recommended because of neuroscience showing that even though these serif fonts are very popular for the written word, like Times is very common in novels that you read, they're very hard to read when projected. And you should try to vary your font size in a range of about 18 to 36. There's also research on that. So no smaller than 18, which I have there for the smallest labels that you have. The only exception might be something like, citing the paper that data came from, that is really kind of for completeness sake and not something that you're asking or you're going to discuss as part of the input of the slide. But try to use that range for slides. Avoid using all capitals, like in this tweet you see here. We kind of recognize that as angry. If you get capitalized words in an email, somebody might just be trying to emphasize or point something out, but your initial impression is that this person is angry. There's no reason to be angry in an invited presentation. Also, there's work to show that a sentence style works best for the slide title rather than capitalizing every word in the title, which I actually did for the first 15 years of my career and then stopped doing that. I mean, you see that in the journal article, but on slides, it works better in a sentence style for reading. And a really key thing for all talks is that each slide should just make one or two points. And the title of the slide is always going to be something that conveys the bottom line of the slide. It doesn't explain what you're about to see in the slide. It explains what you're supposed to understand after the presenter has gone through the slide. And I'll just give you an example from data in a talk I was giving a few weeks ago on a vaccine that we were developing. So this would be a title of a slide that I think would be that you commonly see, but is not a good title. Like I'm describing what the experiments are on the slide. And it's very common that you see slide titles like this. This is an intratracheal challenge of mice with MRSA, and then treatment with red blood cell coated nanoparticles versus placebo to look at the effects on mortality and bacterial load. That's what you see here. There's the model on the left. Here are the results in terms of protection against mortality. And here are the results in terms of pneumonia. But a better title for this slide would be, what do I want the audience to take home from these data? And in that case, it's that these red blood cell nanosponges are helpful in the treatment of MRSA pneumonia. So that's my bottom line. So then I'm going to try to convince people by going through the data, explaining the model, showing how 2 different doses protected the mice against mortality, and explaining how there were different reductions in the bacterial load of 4 or 5 logs in these animals that were treated. And then this shorter title might give you enough to finish up the slide. In this case, we gained FDA approval to move this into human studies based on this and the safety data. Audiences, and I think all of you know as a learner, can absorb information in 3 different ways. You can read what you see up on the slide. You can gain information visual. Many of us are better visual learners, and they can gain information by listening to you. And ideally, you want to have slides in which each one of these elements is being deployed to as a synergistic effort to convey the information in your slide. So if you have too much text, if you lack a graphic, or if you're hoping that the text or the graphic completely conveys the image without you having to explain it, it's not going to be as effective. So you see this all the time, too much slides or 3 text slides in a row. It's very difficult. The natural inclination of the audience is to start reading what's on the slide. At the same time, you're either going to be reading it verbatim, which sounds like you're not familiar with your own research, you need to read it, or you're going to be paraphrasing it, which is very disconcerting because they're reading and you're paraphrasing at a different kinetic. You might be on different parts of the slide, and that's very cognitively dissonant. So you never want too much text on a slide. And really my recommendation, or what has been recommended to me, is a maximum of 2 sentences per slide. And if you're making a list of short little clauses, have a maximum of about 4 items in your list. And often have the list come in sequentially, unveil 1 item at a time on the list so that the audience is brought to that topic simultaneously with when you're ready to talk about it. Don't import whole figures and then ask the audience to focus on one little piece. You see this all the time in presentations. I think, this is a figure from a paper that I was involved in, it goes to panel W on a figure. Okay, this is sometimes thrown up there. Look how much work we did. Look how, beautiful and consistent our data are. But I really want you to focus in on this. This is a very, very common thing. And I think that just shows that you're lazy, or it's actually disrespectful to the audience that you're asking them to do this because of your interest in, showing what a big project it is. Often, people say, to make a long story short, yeah, that's your job up here. You're giving a talk. Your job, we know you did research for a year. You're going to give a talk for 15 minutes. Your job is to make a long story short. You don't have to tell us that. So that this very same data could be pulled out, into a slide, and that's all you're going to talk about, right? I think people understand that there's a lot more work that goes into things. There's collaboration. Obviously, the papers came together, but you're telling a story and only focusing on one thing. Pull it out and show it to us. Keep it super simple. And honestly, I try to have an image on every slide. I just feel like there must be some image somewhere that represents the point I'm trying to make. Here's a kind of Zen stack of rocks, which I think exemplifies simplicity. Once the audience views the image, then they can come back to you and pay attention to you and the point that you're trying to make, because you have the valuable information. You know what you're talking about, and that's exactly what you want. You want them to kind of clue in on something, kind of be excited that the next slide is going to have an image too, and not another bunch of text. Don't use PowerPoint animation effects gratuitously. I study multi-drug resistant pathogens. You don't want them coming in a million different ways and spinning around. And this has no value. These things could have been all in a static slide at the beginning and conveyed the same thing. There are a few things in which animations are useful. Sometimes you want to present data in a staggered fashion to emphasize a point, like antibiotic resistance is a big problem. There are 650,000 annual deaths on average from influenza. We know that so far 6.9 million people have died of COVID. Cancer annually is associated with 9.6 million deaths globally. Antibiotic resistance only is associated with 1.27 million deaths, but on its current trajectory, it will exceed cancer by 2050. So in trying to walk people along through this data, I had the graphical elements come in. You can also do that with slides like this. You can say, here are the leading pathogens. Currently, there are this many million deaths attributable to resistance, but even more deaths associated with resistance in some way. So allowing the data to come in piece by piece allows you to tell a narrative and the people to walk through the data with you. Another thing is to use, what in PowerPoint is called a build. You usually want it to start, either in the left or the upper corner to break down complex experiments into steps. Like here, we were making a vaccine on a nanoparticle from the membrane of a bacteria. So I talked about purifying the outer membrane vesicles, then we put it on a nanoparticle, then we immunized mice and rabbits, then we did the infectious challenge, and we measured the following parameters. Obviously, I would go through that much slower in a scientific presentation, but you can see how it was easier than if this whole thing appeared all at once and the viewer was trying to figure out where to go. Mark Twain once said there are two types of speakers, those who get nervous and those who are liars. And I think everybody has a little bit of anxiety. I've never talked on this subject, for example, so I'm a little keyed up. And you want to channel the nervousness into some dynamic energy about the topic, about your enthusiasm about the topic. One thing that you have to keep in mind is that the audience wants you to succeed. I mean, the audience has sometimes paid to be there at the conference or traveled to be there at the conference. They chose your session over another session. They want you to succeed. No one is looking to say, gotcha, oh, haha, look at the way he blew that presentation. So that should buoy your confidence. Speak about what you know and try to do it in a clear, audible voice, and smile and look at the audience. And, as many people who work in the field of anxiety disorders know, sometimes just acting relaxed can help you feel relaxed. So you have to start to walk the walk, and then that can often perpetuate itself in a positive way. You want to understand that it's harder and harder the bigger the audience, that you're trying to talk with the people. In longer presentations, especially these kind of 50-minute talks that you will give as a visiting speaker and a job talk, you can invite them to interrupt you, ask questions, and in general, you want to scan the room with your eyes and let them know that this is a conversation. You're not talking at them. If you're losing people in the audience, you have to find a way to bring them out. This is from The Art of Manliness, by the way. I'm not sure this website had probably the best slide for that. You want to share like enthusiasm, like I'm passionate about what I do. I care about the problem I'm studying. I'm excited about the potential impact of our research, but you never want to be, preachy because your job is not to say that your type of research is more important than everybody else's type of research, or there are some areas that have political angles. There are some areas that people might be a little bit uncomfortable. There's no reason to be preachy. Be enthusiastic. Many people will join you. There'll be a few people in the audience, every audience for which your topic is not the most exciting topic. That's fine. And then I just want to wrap up by saying, like, the general structure of a presentation is the same as with a paper, this kind of hourglass model where you want to start out general and broad and then narrow into the specific information. That's where you'll get into the nitty-gritty of your research study, and then bring it back out broad. So you can kind of think of different episodes over the course of your talk. Usually you'll be starting about what is the problem that you're talking about scientifically. Let them know what the unanswered questions are. Then you get narrow. How did you specifically approach the study? What were your results of your experiments or clinical study? And then end broad again. What did you learn? How does this change things? And where do we go from here? And just along these lines, I'm going to show you a couple of slides that I came across that are more kind of metaphorical, or they have a graphic element that I find I keep pulling into various talks. I've been doing them for like 10 years, and they seem very versatile kind of slides. And every one of you want to sit down and think of some slides like this. Like when I'm describing what my lab is, instead of a list of things, I tell them, I study the way common bacteria interact with the innate immune system. And I talk about how the innate immune system is usually winning because it's very strong and formidable, and we have bacteria all around us and don't get infected. But occasionally bacteria break through the immune system to produce disease. And we call those aspects virulence factors. And this David and Goliath analogy fit well for that. This famous video that many of you have seen of a neutrophil chasing down a bacteria is a very good analogy for me about the evolutionary dynamic over time between bacteria and the immune system. We're trying to eliminate the bacteria, but they're trying to survive. And even when they get phagocytosed, like you see in the video, they can still survive intracellularly. And we're dividing every, 25 or 30 years or 40 years, if you waste a lot of time in medical school and stuff like me. They're dividing every 20 minutes, and they have this tremendous advantage. I wrap up my talks often by going big, saying, this is how we currently study antibiotics just based on lab tests, but we really need to think about personalized medicine, where we're asking, are our patients getting better? Are our patients getting worse? It doesn't matter what the lab test is saying. If your patient is receiving the antibiotics and they're not getting better, the bacteria is antibiotic resistant, and we need to learn more about the immune system and new types of therapies. Otherwise, we just keep stepping on the same rake because this problem is becoming worse and worse. And the last slide I like to show is talk about me as an infectious disease physician, and all infectious disease physicians, how we became complacent. We relied on antibiotics, but now the bacteria are becoming resistant, the patients are becoming more complex. And I say, how about the cancer doctors? They never had that luxury. There were so many cancers that never had a good treatment. They were always searching for new ideas, and they came up with magic bullet type of drugs like Gleevec, which targeted the specific kinase of a particular cancer. And that had huge outcomes in chronic leukemias. And now we have immunotherapies that are working through the host to fight the tumor. And we could have these same advantages in infectious diseases. And my last slide talks about going back to what I study, that host pathogen interface, and pointing out that right now we only have classical antibiotics, but if we targeted the host pathogen interaction, there's all sorts of new drugs that we could discover. So use graphical elements you bring it full circle to something you talked about. You kind of think, give a vision of what I think the future is in this field with a little bit of an appeal linked to that model that cancer therapeutics has shown for us in doing stuff. So sorry for the AV glitch at the outset. That's probably another point I could have made, come a week early to your talk and make sure everything is rolling, but always rely on the brilliant saves of your AV crew. And if there's any time, I'll be happy to answer questions, but I'll also be here all day today and tomorrow to pull me aside during any of the times. Do we have a question or two? This is why you don't put in any questions. There may not be any questions. That's a recommendation I have for everybody. It's just a nice comment from Rupsa. Thank you, Rupsa. Any specific sites you recommend for biology graphics? Yeah, there are a number of public domain sites. There's also a number of sites in which foundations like the American Society of Microbiology has a public image library that they've created. Obviously, anything in Wikipedia is in the public domain. But if you're going to be giving a lot of presentations, it really doesn't cost that much. For example, $25 a year or something to get associated with, some giant, picture libraries. I'm not going to say, Getty Images or something else because I don't know the pricing on there, but there are some very large sets where you could literally type in anything like, I need a kid with a thermometer in his mouth and a rash, and then there's 4 or 5 kids models posing in that settings, and that the image is free for you to use in academic presentations like this. For drawing images, recently I've started using BioRender a lot, which I think is a very useful tool. I can see a few nods from, especially if you're doing scientific images. I think it's very, very powerful. You can import any image that you have from the outside world into BioRender too, and then they have a huge library, especially of different kinds of cells. And I need a mass spectrometer and an example of a Western blot and a patient with an IV. These things will quickly come into your presentation. You'll recognize the images immediately because of how widely it's being used. I mainly use PowerPoint and Illustrator and then BioRender online. I don't know, anybody else have a recommendation of a tool they use to put images together? ChemDraw has some biological pictures too, Lindsey says online. Okay, I will stop there because there's going to be a lot of great talks coming up.
Thank you, Dr. Nizet. So our next session is a series of flash talks that will be presented by a number of trainees and junior faculty. So for this session, we have 5-minute talks from each person, and we will be cutting you off after that 5 minutes to give time for the next person. Sorry, let me just try to find them here. And then after these 5-minute presentations, we'll have a short break. And then for those who are in person, we have breakout rooms assigned to you for those who are giving talks. And then some faculty and other attendees have already been assigned rooms. If you have not been assigned a room, you're welcome to go to any room or stay in here where we will be giving feedback to our virtual participants with the goal of really, taking what Dr. Nizet and Dr. Sarah Van Dries, who talked a couple of weeks ago in one of our webinars, taking, their advice and adding to it, giving feedback on presentation and communication skills to our trainees. So, all right. Looks like we are not extending correctly, so. Okay. Maybe we're going now. All right. With that, we will invite our first speaker up, Dr. Nina Zhao from the University of San Diego. Nina is in the Peter Dorenstein lab and will be talking on empirical medication readout and untargeted metabolomics. So Nina, if you would like to
Thanks for the introduction. Good morning, everyone. It's a really great opportunity for us to be here, present our research, and receive feedback on our presentation. Hopefully, I have all slides with a white background and the black font, so hopefully that will not annoy the audience. I'm Nina. I work in the Dorenstein lab as a postdoc here at UCSD School of Pharmacy. And today I'd like to share a mass spectrometry drug library I recently developed with you that I hope will be very helpful for medication readout in non-targeted metabolomics research. So human get exposed to a lot of chemicals in our life. Beyond the chemicals we get from food, from environment, from microbe, medication, in my opinion, is a very important source of chemical exposure because those chemicals was designed to be bioactive, and they can have a very large impact on the biology of human body. So right now, to study the relationship between medication and disease, we usually rely on the medical record of the patients, or we do some kind of survey during the clinical visit. However, those information, those data are very expensive and very hard to get. And sometimes they were just not available. And even if those data, those clinical records were available, they do not consider the different metabolism of each person. They also do not cover medication exposure from other routes, such as from food, from the environment, and also people were very creative at getting drugs. They might just get illegal drugs, or they also have like some kind of like online wandering source. So overall, we think we really need, because of these reasons, we really need a better or more accurate way to evaluate the medication exposure of patients. And that's what I want to work on in this study. We think non-targeted metabolomics can really help the situation here by actually measuring the drugs in human body. So my job here was to develop the necessary tool that allows medication readout in non-targeted metabolomics research. So the most reliable way to annotate or to detect the chemical in metabolomics research is through library matching. So here, I want to develop a comprehensive mass spectral library for drugs to facilitate drug detection in metabolomics research. I'm not going to waste my precious 5 minutes on boring technical details, but basically, briefly, I acquired a lot of spectra, mass spectra for drug from public mass spectral library, and beyond that, I also collaborate with Dr. Karina Brox at Czech Republic. We measured some new chemical standards to acquire mass spectra for those new drug or new chemicals. These efforts gives me a drug spectral library that has 100,000 spectra in it, belonging to roughly 5,000 drugs. So roughly we have 20 spectra per drug. The beauty of utilizing public library spectra, instead of me measuring all the chemical standards in one instrument, the real advantage of acquiring all the spectra from public library is that those spectra was contributed by the community. So they were measured by different labs using different instruments, using different methods. So that collecting those public spectra really allow us to have a broader coverage of reference spectra under different conditions. However, drug will be metabolized in human body. So we want to cover not only the parent drug, but we also want to cover their metabolites in our library. So to achieve that, we utilize the mass spectra in public metabolomics database. I'm not going to go into details here again, but we basically developed some computational tool to capture the spectra that's related to drug in the public library. We were still trying to improve the tool, but so far I have been able to capture 10,000 spectra for drug metabolites through this public domain. I know my time is running short, but just briefly, to apply the library, we screened drug in 500 human breast milk samples analyzed by Dr. Sydney Thomas, which I think you will hear in 20 minutes. We were able to detect 35 medication and some of the most frequently observed one, including some antidepressants, anti-epilepsy drug, as well as pain relief drug. So that agrees with the medical usage of the breastfeeding and the young cohort. So we really think this non-target metabolomics approach coupled with my drug library can help with not only the research, but also clinical practice in the future. So I'd like to thank everyone in the Dustin lab for their help in the project. And we were, as you can see, we were still in the early stage of this development and we were looking for collaboration. We were always looking for collaboration. My email is there. If you want to measure drug, just let me know. Thanks.
All right, good job. Great timing. As soon as it was finishing. So our next speaker is Dr. Dustin Huynh, also from UC San Diego.
Hi, everyone. So I'm going to be presenting on the infant and adult VRC07 population pharmacokinetic model to basically predict the HIV prevention in breastfed infants. So a little bit of a quick introduction. My name is Dustin. I am PharmD and I went to UC San Diego School of Pharmacy and I graduated back in 2022, so very recent. And currently I am a UCSD and Pfizer Clinical Pharmacology and Oncology Fellow here over at the La Jolla branch. So a little bit of background about my project. There is a difference in the success of prevention of mother-to-child transmission of in resource-poor and resource-rich countries. And there's actually a partial list of factors that could include the late diagnosis of HIV infection, the overall lack of universal access to highly active antiretroviral care, the continued need for mothers to breastfeed into over 1 year of the child's life, limited access to maternal as well as infant antiretroviral therapy, and then just overall the lack of adherence and even maybe a contraction of a resistant HIV virus. And this presents a potential need for a new novel therapeutic that can be used prophylactically or just used as an intensive regimen early on. So the previous phase 1 and phase 2 clinical trials of a broadly neutralizing antibody actually demonstrated favorable safety profiles as well as pharmacokinetic profiles. And currently there is a need for bNAbs that are have longer half-lives as well as higher potency, because we want to make sure that the antibodies are staying in the body long enough to prevent HIV. Right now, there are lots of routes of administration. Current antiretroviral therapy is mainly oral, but we're looking into having routes of administration that are more subcutaneous based or intravenous based, just so that we can address these adherence challenges that were presented previously. And combinations of bNAbs are currently the landscape and potential future of designing bNAb regimens, as we want to target a lot of the different epitopes on the viral envelope of the HIV molecule. And this may be the key to having prevention efficacies that are high enough to prevent HIV early on in the early years of life. So the bNAb of focus today that I did my research on was VRC 523 or 523LS, which is a novel second generation broadly neutralized monoclonal antibody that actually targets the CD4 binding site on the HIV molecule. And in comparison to the generations before, it displays a greater breadth of coverage as well as neutralization of all of the viruses, clade B and clade C that it was tested against. And as of right now, in current research, it's safe and it's well tolerated at plenty of dose levels as well as routes of administration. So for my methods, I had pharmacokinetic data from two separate studies, one study being healthy adults and the other study being HIV exposed infants who initiated the therapy at less than or equal to 5 days of age. And it was a 2-compartment model that was done in non-mem that used allometric scaling. And I had 683 concentration samples that were collected using ELISA, and covariates were essentially evaluated on a univariable basis and then moved on to a multivariable basis. Once my model was finalized, I had run Monte Carlo simulations of the final models and assessed 2 doses. I assessed the dose at 80 milligrams at birth, and then another 100 milligram dose at week 12. And this was to determine whether or not VRC07 by 23LS serum concentrations were above our target level of greater than 10 micrograms per ml. I also introduced a surrogate biomarker, PT80, which incorporates bNAb concentrations in addition to IC80 levels. I have a calculation down there, but basically the calculated PT80 values were essentially used to predict or estimate HIV prevention efficacy as a percent value. And this was used from a bNAb pharmacodynamic model that was done previously in an AMP study done by Gilbert et al. Oh, man, thank you. Oh, went way over, sorry.
Thank you, Dustin. So for Dustin and those who don't get time to go through your slides, that's why we did this at the beginning, and you will have plenty of time to discuss your projects throughout the rest of the conference. So, all right, our next speaker is Dr. Aislinn O'Kane, who's a PharmD T32 fellow, clinical pharmacology fellow at Indiana University School of Medicine, and Aislinn is joining us virtually today. So Aislinn, are you able to speak?
Yeah,
I can hear you. Awesome, great.
Okay.
All right. Go ahead whenever you are ready.
Thank you, Sarah, for that introduction. I apologize if this is a little clunky as I am presenting virtually to you, but my name is Aislinn O'Kane, and I am about 9 months into my clinical pharmacology fellowship at Indiana University, and I'm working on the development of a physiologic-based pharmacokinetic model for dexmedetomidine, brand name Precedex, in pediatric patients. So a little background, dexmedetomidine is an alpha-2 agonist. It's IV only. It's basically like the IV version of clonidine, and it's used for the initiation and maintenance of sedation in the ICU. It's not currently approved in pediatrics by the FDA, but it is used very commonly off-label in pediatrics. At our institution at the Riley Hospital for Children, pretty much all patients that you see that are in the ICU who are intubated or sedated are going to be on dexmedetomidine, and it is recommended by the Society of Critical Care Medicine as a first-line agent for sedation in pediatric postoperative patients with early expected extubation. It has a very high dosing variability. Dosing can vary between 0.5 and 1.2 micrograms per kilogram per hour, which is titrated to sedation scores. This unfortunately leaves a pretty big window where patients can be either over-sedated or under-sedated, and both of these have their own risks. So under-sedation can lead to self-extubation, which is pretty common in pediatrics and can be very harmful, and over-sedation can actually lead to delirium risks, even though dexmedetomidine has a lower delirium risk than most benzodiazepines. You can go on to the next slide. Thank you. So for our methods for this model, we collected literature pharmacokinetic data. This was mostly taken from the FDA label and the clinical pharmacology review, as well as the EPAR. This included the log-P, blood to plasma ratio, clearance, pKa, volume of distribution, and then also some genetic data from a recent study done by Kaibo Seri et al., which quantified the glucuronidation pathway of dexmedetomidine. And we use this to build our healthy adult base model, which we are then going to validate against adult literature data. And hopefully this will be within twofold of that adult literature outcomes. And if it's not, we can go back and we can refine the model based on that. And then once we have our model validated, we can move on to scaling it down to pediatrics. So we're going to simulate it. And this is in SimCip version 21. I forgot to clarify that. So we will then simulate it in the SimCip pediatric population in SimCip version 21. And we will again validate this against literature data. We're very fortunate in that many population pharmacokinetic studies are coming out in pediatrics on dexmedetomidine, which is really exciting for us and exciting for the building of our model. And once we validate this, it's our hope that we can use this model to help predict what the genetic effects are of dexmedetomidine in patients, as well as the effect of ontogeny in patients as they are getting older and their genetics are changing. So our project goal is to use this PBPK model to optimize the dexmedetomidine dose in pediatric patients, accounting for both genetic variation, so this includes the glucuronidation pathway, which has been very well defined, as well as the CYP pathway, which has been a little less defined right now. We're actually still working on that, and we may be doing some in vitro studies at IU to help quantify this and feed the model as well, mostly CYP2A6, CYP2D6, and CYP1A2. And then also figure out the effects of ontogeny on how dexmedetomidine is metabolized in patients and how it's affecting their pharmacodynamics as well. So for our progress, our base model is built. We're still working on the genotype model a little bit. We're still trying to exactly figure out how much of dexmedetomidine is going through each CYP pathway. We have a really good idea of the glucuronidation, but we're still working on that CYP metabolism. And then next is going to be our adult model validation. So verifying our model against adult literature data and making sure that it's accurate, and then scaling it down to pediatrics and SimCip, and then validating that against pediatric data and assessing our outcomes. What questions do you have for me?
Talks. And then if there are questions, feel free to reach out to Aislinn or stay in here for the breakout session, and you can ask her during that time. So, all right. Our next speaker is Dr. Sydney Thomas, and Sydney comes to us from the Dornstein Lab here at UCSD.
Hello, everyone. My name is Sydney Thomas. I'm a postdoc in the Dornstein Lab, and today we're going to be talking a little bit about human milk. So human milk has had kind of an interesting last century or so, where it went from being really the only safe option available to new parents to being almost completely replaced by infant formula. A change brought about in part because of this kind of widespread idea that human milk was somehow deficient in supplying the dietary needs of the infant. But while it's clear that access to safe infant formula remains very important for many parents and infants, we still have not been able to create an infant formula that in any way recapitulates the health benefits of human milk. These benefits begin at day 0 and continue throughout childhood and even into adulthood. But while it's clear that exclusive breastfeeding is incredibly beneficial to both the parent and the infant, we still don't really understand how milk is achieving all of these positive outcomes. We do, however, have some hints of specific components in milk that are very important for infant development. The first of these components are human milk oligosaccharides, or HMOs. These are sugars that are specifically produced by humans but are completely indigestible by the infant. Instead, they provide the main source of fuel for the infant's developing gut microbiome. However, HMOs are not the only way in which human milk can influence the infant's gut microbiome. Human milk itself is not sterile. It has its own complement of live bacteria, and it looks like some of these bacteria can actually directly colonize the infant gut as well. Now, of course, I'm a metabolomics person, so I would be amiss not to talk about the human milk metabolome, which is incredibly diverse and dynamic and can change on orders of abundance within a couple of hours to a couple of days. So with all of this, we know that each of these individual components are very important to to infant development and parent health, but it's unlikely that any individual component is going to answer all of our burning questions about human milk. So instead, we took a multi-omics approach. To do so, we collaborated with partners across the globe to analyze around 800 human milk samples via 3 omics methods: milk microbiome sequencing, untargeted milk metabolomics, and human milk oligosaccharide analysis. Now, once we had done this, one of the first things we were interested in looking at was how levels of different individual human milk oligosaccharides affected the milk microbiome. We know they have a huge impact on the infant gut microbiome, but we don't really know how they impact the milk microbiome. So here's what that looks like. We got a lot of individual human milk oligosaccharides here on the right, and their correlation to the alpha diversity of the milk microbiome on the left. You can see there's a lot of geographic variation here. But what was really interesting to us about this result was that there were 4 HMOs, 6SL, DFLNH, DSLNH, and LSTC, that were not only very negatively correlated to microbiome diversity, at least in the US data set, but were also positively associated with several parental health outcomes, things like C-sections, preterm birth, as well as the use of over-the-counter pain relievers and SSRIs. So we wanted to kind of dig into that a little bit further and look at if there are any specific bacteria that were associated with these HMOs, these 4 specific ones. And so this is a big analysis of how this works. And what you can see is that there are many individual bacterial genera that are associated with these 4 HMOs in one data set, but there's only a handful that are associated in multiple data sets. I'm going to list them out here. What's interesting about those bacterial names is that in general, the bacteria that we see increasing with increasing amounts of these HMOs tend to be bacteria that are associated with the skin microbiome, while those that are decreasing tend to be associated with the oral microbiome. And since we know milk microbiome samples often contain microbes from the parent's skin as well as the infant's mouth, we hypothesize that this might actually have to do with feeding differences between high and low HMO producers. Now, to figure this out, we actually have to categorize what is high and low production of these 4 HMOs. This was harder than it sounds, but we were able to do it. And once we did do this categorization, we didn't see any significant differences in kind of the normal covariates you would think of: parent age, infant sex, etc. However, we did see a small but significant decrease in the rate of exclusive breastfeeding in high HMO producing parents. Now, what was really interesting about this result is while we saw a decrease in the amount of exclusive breastfeeding, we saw an increase in breastfeeding frequency. So the y-axis here is breastfeeding frequency in times per day. We have parents in this cohort who breastfeed up to 24 times a day, which, I mean, pour one out for them. But the problem is, if you're breastfeeding that much, that generally indicates that you're having some sort of difficulty breastfeeding. The infant isn't latching, you feel like you're not producing enough milk. And so what we're seeing here is these parents are trying really hard to successfully breastfeed, but they're also more likely to fail. What's interesting is there doesn't seem to be any physical issue, and I will be done. But first of all, thank you to all the people who helped collect this data. Sorry, I missed my, thank you, Sydney. Sorry, I missed my 30-second warning for you. So
All right, our next speaker is Dr. Mohammed Yaseen Abbasi, who's from my lab at Indiana University School of Medicine. Try to fix this issue. All right.
Thank you very much for this opportunity. And 5 minutes is like too much late for me because I've been working a lot for this model. So just I want to start with my acknowledgements, and these are my people with I'm working, and I'm working on building a lactation model, gabapentin, for the especially for the how much drug is going. Gabapentin is reaching to the neonates via when they are on the feeding on a mother. So gabapentin is like a hydrophilic drug, and the pharmacokinetic data of the gabapentin is very low, and especially for the concentration in the pregnant woman and the milk concentration. So there is no food effect for this drug. Half-life is very small, and there is no metabolism for this drug. So when we are discussing about the breastfeeding for the drug, there are some criteria that that drug should have a relative infant dose less than 10%, and the milk concentration, milk to plasma concentration should be less than 1, and the binding should be very high, and molecular size should be less than 200 Dalton. So, and poor viability and metabolism as well, and half-life. So if this is the criteria, then drug should be passing to have a we can go for, but we cannot say exactly. So these are the criteria. So my objective for the gabapentin is that how can I build a PBK model, and how can I predict or calculate the infant daily dose and relative infant daily dose. So for this, I use this SimSip as a software, which is very good to predict the a lactation model, like relative infant dose and infant daily dose. First, I have validated my data in the healthy volunteers. Then I went for the healthy volunteer females, because my work is to predict the concentration in the female and the milk. And then I have went to use the lactation model in the PBPK. But the problem is that I have to create the scenario, especially for the early postpartum pregnant woman. So that is very tough, but I tried. Because in clinical setting, we don't have data, what the physiological changes are there, and especially women having a C-section, that is a critical issue. So these are the simple things that I have used, that everyone who is working on PBPK model, they need to know these physicochemical properties and pharmacokinetic data. And when I build a model for the healthy volunteers and the healthy female woman, then this is my data, and it looks like good, that it is predicting well. So I got a confident. I went further. When I was changing the physicochemical properties, then I got few data that I am using. Like I changed the GFR value, and I use the different 3 kg as a body weight for the infants, and I assume that on the first and the second day, infant is taking 50 ml of milk per day. But I assume. So we have 2 studies to validate. One is a case study, and one is a clinical study, which is in Indiana University School of Medicine we did. So we have a different 2 doses, like 300 QID for 48 hours and 600 TID, and I have validated this data. When I validate this data, so for my case study, which is only in 1 patient, that my model is predicting very well. But in the gabapentin clinical study I did not predicting very well, and you can say my ratio is very high. So this is due to the physiological changes that I am not able to capture very well. And in clinical, in literature, I'm not able to find the physiological changes. So I'm looking some way, because recently few papers has published like Andre Delman, but they have worked on the PKSIM, but I'm working on SynSIP. So I have to transfer that data. So I'm working on it. So maybe in future I can predict well, or try to change the physiological and capture well. So I believe that this challenge is due to different physiological variability, like multiple drugs, like co-administration, physiological changes, races, different race, and fluid intake and the surgery. These might be the reasons. So I can say the case study, which is in late postpartum, This is predicting well, but for the early postpartum, my model is not predicting well. We need a clinical studies or some data so we can do that very well, or we can capture the physiological changes very well. So thank you very much.
Thank you, Yaseen. Our next speaker is Dr. Simone Zuffa from the Peter Dornstein Lab.
Hello, hi everyone. My name is Simone. I'm a postdoc here at UCSD, and today I will present a tool, a mass spectrometry search tool that we have been developing in our lab. I guess if you are here today, you are quite familiar on the concept of how the gut microbiota has a huge impact on human health. There is a huge genetic potential expressed by this microbe that they can produce also thousands of different molecules. Some of them that are extremely important, like energy metabolites, vitamins, or neurotransmitter. But what is very interesting is that we have many unknown molecules that can be produced by this bacteria. And a way to study this molecule is using mass spectrometry that Sydney and also Nina talked about you before. Basically, in our lab, we use untargeted metabolomics approach to discover novel molecules of biomarker, for example, in case study where you have healthy patients versus patients with specific diseases. So when you perform an untargeted metabolomics experiment, what you get out of it is basically big tables with 10,000 of variables. And if you are a data scientist like me, what you do as soon as you get your data, you throw everything that you have at it. You start building all kind of model, PCA, PLSDA, random forest, mixed effect models, and then you get like perfect models that are able to separate your control from the disease, and you are extremely happy. But then what? You look at the top 100 molecules that are separating your two groups, and they are all unknown molecules. And then you start crying because it will take months just to understand one of them and try to annotate them. So that's the big elephant in the room. Untargeted metabolomics is an extremely powerful tool, but 90% of the data that it returns is basically unknown. And we have no idea if these molecules are coming from drugs, as Nina previously mentioned, if they're coming from food, if they're coming from the environment, or if they're coming from the gut bacteria. So this is the question that we had. Can we tell if a known molecule that we observe in untargeted metabolomic experiments are coming from bacteria or fungi? So we had the idea to query public available data in order to understand and propagate this information and see if we can tell if an unknown molecule is produced by this microorganism. And this is, in a nutshell, what microbmast is. We scraped public repository like GMPS that was previously mentioned, and we were able to extract monocultures of microorganisms, specifically of bacteria and fungi, and we had more than 60,000 experiments that comprising 100 million MSMS spectra, so possible molecules. We then built a tool to quickly go through this spectrum and look for association, and we associate metadata available to all these spectra in order to build taxonomic trees that can be returned as a result of your search. So I'll quickly show you the web interface of MicroMast. What you can search is basically your MSMS spectrum. You can just put your fragment ions and the intensities, or otherwise you can just add a universal spectrum identifier that is like a way, like a UR link for mass spec. Let's see. And then you just click search, and what it returns is like a interactive taxonomic tree of where you can observe where the molecules of your interest was found in the specific monocultures. I'll just give you a brief application. So you might know that all these molecules that are influencing health, some of them are extremely important that we are working on are the bile acids that may play a role in the inflammatory bowel disease. We recently discovered new colligated bile acid in our lab. One of them is phenylalanine acid. And when we were studying these molecules, we observed that it was in higher prevalence in patients with chronic disease. And we also observed that when people were treated with antibiotics, this molecule was actually decreasing. So we came up with the conclusion that this molecule would actually be a microbial derived molecule. So the question is, which bacteria can produce these molecules? Well, we searched these molecules with micromast, and we were able to observe that several bacteria that were already previously associated in literature without DB were producing these molecules. So giving a hint of how micromast can actually help you understand causality and design more mechanistic experiments. Micromast is already available online, so you can scan this QR code if you want to try it. I hope I caught your attention. If you want to chat with me during these days, please come to talk with me. Thank you.
All right, and our next presenter is Dr. Lijun Chen, and Lijun will be joining us virtually. So Lijun, are you able to, are we able to hear you, I guess is the question.
Hello, everyone. Good morning.
Morning. Okay, great. We can hear you just fine. So Lijun Cheng is a assistant professor of biomedical informatics at Ohio State University, and she's going to talk about a project she's working on, potential target prediction in preeclampsia. So Lijun, the floor is yours.
Thank you. Thank you for introduction, Sarah. I'm so happy to share with you guys on my research in the preeclampsia, especially for severe preeclampsia, the drug development. So I focus on the current use of the bioinformatics. Can I share my screen? Okay. Great. So I use the bioinformatics strategy, start from the multi-omics data and identify potential target, and try to do a translation and start from the human and the mouse connection, and did an experiment validation and the final for the clinical trial. So my study is how can I set up a pipeline? Those pipeline is based on the bioinformatics strategy and contact the different type of the experiment, start from human and the mouse model and until to the clinical trial for translation, and try to quick this step from the drug development. I use the preeclampsia as one example to show this. This pipeline is try to the current in the severe preeclampsia and put it in the possible and for translation in the future. So we start from the mRNA data and use next generation sequence testing from the patient generally, and then use the different pathway and analysis, look at which there is a disease gene and where is got wrong and cause the preeclampsia happen, especially for severe preeclampsia. And for those target and potential disease gene, and we look at which the mouse model, which the mouse can help and try to do experiment and validate what we find. At the same time, look at the drug development. The last part, we use the literature review and look at the potential those drugs if it is possible and for clinical trial, and at the same time look at biomarker identification. Next slide. Thank you. For preeclampsia, the background information is that is a multi-system disorder that affects 3% to 5% of the pregnancy ladies. So a lot of the child and the ladies, when they got the preeclampsia, will cause the organ damage and will cause the baby die. So that's the reason we will look at if we can do a prevention, or for severe preeclampsia, if we can identify the potential, the drug can stop those, processing. Next slide. Thank you. So but the current is still lack of the treatment for severe preeclampsia, and it is well laid a very serious, even fatal, complicated for both of mom and baby. So that's lack of the treatment for postpartum preeclampsia. even like those baby after delivered, those moms still have the symptom and are very dangerous in their the current, the healthy manage. Next. Yeah, thank you. We want to look at how they cause the preeclampsia. So we use a big data analysis. Here, give example. We collected the data from their GEO gene omnibus expression data set, and that is all related to the tissue placenta, and there is well touched their control and the preeclampsia group, and in the early stage and the later stage, and the severe preeclampsia and the control group, and from the phenotype connection with the genotype, and identify the specific, the potential we call disease gene and as a target. Here we use the method. Please go to the next. Yeah. We use the method, and here we give example GSE 75010, and there is a 29 different the clinical phenotype, and we integrate with the genotype, the gene expression variation. So there's 157 different samples. Next. In the preeclampsia, the severe preeclampsia, the liver damage is very, as one type of the typical syndrome. So we use a linear model and to look at the score and across the different phenotype and identify the specific their disease gene, those genes across the symptom, liver damage and their urine protein, and like the others, like high pressure. So here I show some example is 844 genes that is we find those genes are very associated with the disease that's caused in the preeclampsia. Next. Please next. Yes, and after that we try to look at associated the target and get the target identification and the drugs treatment and the validation on the mouse. So we would like to find more validation in the clinical trial and others, hopefully with other persons. Can you understand? Can you hear me? Yeah, that is all what I'm working on.
Okay. Okay. Do I go back or start? From the beginning? Okay. So today I'm going to talk about my current first year graduate program project that I'm working on. It has to do with monoclonal antibody Fc fusion drugs in pediatric care.
So there's currently 140 approved of these drugs approved in the clinic. And of that 140, I am currently working on 20 that can be used in pediatric care. Of those 20, there are a couple that have no pediatric PK studies. And what I'm going to show you guys today is just how I developed the models, and then I applied them with the digitized data from the literature research. Let's see. So on the, I don't know if I can. So on the graph over here, I have showing the model that I ended up creating from the data I pulled from the literature research. On the graph on the right-hand side is the data that has to go with this graph over here that I digitized. And then I ended up superimposing them and got them to, it showed that the model data that I had came up with did, the model data that I did come up with was matching with what was found in the article. And then I did the same thing for the following drugs. So you'll see in the slides, you'll see the same thing going on. And then some of these are still in progress, so I'm still working on getting them to fit better. But I have the same thing going on here. I have the superimposed graph, the graph that I was able to find data for, the data from the article, and then the digitized version of that. And I'm just going to go through them because it's repetitive with all of these drugs. There are some that I'm still trying to figure out. So some I have modeled, but then what I have from the articles looks similar, but when I do superimpose them, they don't fit as preferred. So there's still of these 20 that I'm still working on getting to work out. Sorry, my presentation isn't as detailed as everyone's, but what I'm aiming to do at the end with all these that I get to have modeled is we're aiming to prepare an app that I'll be able to input all these models into, so then that clinicians can enter the age and the weight of the child, and then it'll easily be able to output their dose recommended on their pharmacokinetic based information. And that's all I have.
Our next presenter is Dr. Caitlin Aamodt from the Nathan Lewis Lab here at UCSD. Caitlin. And she will be talking on using transcriptomics to link metabolic dysregulation to endophenotypes in autism. Over to you.
All right, my name is Caitlin Aamodt. I'm a postdoc in the lab of Nathan Lewis here at UCSD, and our research group is really interested in how we can improve the lives of kids with autism spectrum disorder. As most of you know, autism is can be a very debilitating disease, and it's highly heterogeneous. So one approach that we wanted to take to better understand what might be going on and what might be contributing to specific aspects of autism was to take blood samples from a deeply phenotyped cohort of 363 kids, half with autism and half typically developing. And we performed RNA sequencing to see how their transcriptomes might differ. Then we took this data and analyzed it using a web tool developed by our lab called mSelfie. And the way mSelfie works is that it uses genes that are involved in metabolic signaling cascades and analyzes those genes to see how these reactions might differ between one sample and another, or one condition and another condition. And as most of you well know, what makes metabolism really cool and interesting is that these reactions have been really well characterized to the point where scientists can build models that describe all of the necessary molecules required for those reactions to proceed. And then M-Selfie takes advantage of those models by looking at how levels of these genes differ and describing how they contribute differently to these metabolic tasks. So today I'm just going to highlight a few of our results that were particularly interesting, and then also describe how these results parallel with some of the results we got in other tissues to find converging similarities. So one result that we were happy to see was that we saw reduced glutathione metabolism in peripheral blood, and then we were also able to see that in postmortem tissue taken from subjects with ASD. Glutathione is it's a antioxidant, and it's also really interesting because it's been previously shown to regulate the NMDA receptor, which is really important for higher order cognition. So another reason we were really happy to get this result is that previous studies directly characterizing metabolomics in autism in smaller samples also saw the same change. So it was a good internal sort of validation that this tool is actually relevant for understanding metabolic differences. Another interesting difference we saw was reduced activity of the purine salvage pathway. And this caught my attention because there's a syndromic metabolic disease that causes some behavioral features that are really similar to autism, including self-injury, and of course, severe neurodevelopmental delays. So this was really interesting because it suggests that some of the interventions that are supportive for kids with this syndromic disorder, Lesch-Nyhan syndrome, could also be beneficial for kids with autism. Another interesting parallel to an inborn error of metabolism was reduced oxidative decarboxylation of pyruvate, which in a syndromic form is associated with Leigh syndrome, which causes seizures and neurodevelopmental delays, which are also commonly seen in autism. And in particular, we saw differences in girls with autism, which is what's shown on stage left. And we also saw that this was one of the major changes in induced pluripotent stem cell-derived neural progenitor cells. So that led us to wonder, in cases where a parent is having a second child and the first child had autism, and the parent would be at high risk for having a second child with autism, could we find ways to modulate the diet or do a supplementation that could improve the outcome for the kid? So in conclusion, I've introduced the web tool M-Selfie, which can predict metabolic dysregulation in various sample types. I've shown that glutathione metabolism and purine salvage are downregulated in ASD postmortem cortex and peripheral blood. And I appreciate you all for your attention.
All right, great job, Caitlin. So our next speaker is Dr. Mokshada Kumar from Dr. Deval Shah's lab at University at Buffalo.
Hi, everyone. A very good morning to all of you. I'm Mokshada Kumar. I'm a third-year PhD student from University at Buffalo, and I'm studying right now in Dr. Deval Shah's lab. Today, I'm going to be talking about developing PBPK models to predict the PK of monoclonal antibodies in pediatric population. Monoclonal antibodies are a rapidly growing therapeutic modality in the clinic, and currently we have greater than 140 clinically approved antibodies. Out of these 140 or more, there are only approximately 25 which are approved by the FDA for use in pediatric age groups. Now, this highlights this huge gap between antibodies that are already being developed but are not available for pediatric age groups, and we want to just narrow this gap and make sure that more and more antibodies are available to pediatrics. Now, pediatric populations are divided into majorly 4 stages of development. your neonates, infants, children, and adolescents. It is widely known that while adolescents might have similar PK and physiology to adults, but there are significant differences between the physiology and pharmacokinetics of these antibodies between infants and adult population. And therefore, adult PBPK models or adult dosing regimens might not be as effective in pediatric population. We need to use PK/PD modeling for pediatric therapy so that we can provide better treatment options for pediatrics. We also want to reduce widespread off-label use that is currently present in the clinic because there are not enough options of pediatric drugs. And we want to develop better dosing schedules so that we can gain maximum efficacy. We want to reduce the dependency on pediatric clinical trials because there's a lot of ethical concerns in having huge pediatric clinical trials. And we also want to give a possibility of personalized dosing to these kids based on their age, their body weight, etc., which I will also show you an example in my later slides of. Towards achieving this goal, we have developed a PBPK model, the structure of which I'm not going to go into details for the benefit of time, but I want to highlight that in this PBPK model, we have included equations that have been published by our lab previously, which calculate your blood flow rates and your organ weights, your hematocrit based on the inputs of age, body weight, and sex of the patient. So we keep modulating these. Also in our PBPK model, we change the age of the patient based on time for us, because in chronic conditions, usually you have PK data for 1 or 2 years, and your patient might undergo severe physiological changes during this duration. And so we account for these changes while our PBPK model is predicting the PK of patients. So over here, I have PK plots of around several antibodies, and I want to show that our model is currently able to capture the PK of these antibodies ranging from, as you can see over here in palivizumab, 0.13 years. So patients who are really tiny infants, and also adult patients, like for example, over here, atezolizumab, 12 to 18 years, for both single dosing as well as multiple dosing. Our final aim or our goal with all of this model is eventually to build apps. And I will be showing an example of an app. So you can scan the QR code there to go take yourself to the app that is currently available online if you want to visit that as well. I'm just going to quickly take you through the app over here. So when you open, you'll be taken to a UI, which welcomes you, and you can do 2 basic things. So this is especially for clinicians in the clinic. So if you do not have any previous information on the patient, you can input the body weight, age, and sex of the patient, and you will be able to see the PK. And you will also get some certain outputs based on like what trough level, what % of patients are able to reach those trough levels. And if the clinicians have some previous, let's say, PK data or concentration dependence, so based on that person's individual PK data, the app is going to calculate what sort of dose is the perfect dose to reach a certain trough concentration. So over here, for the ease, right now I've clicked on no for previous concentration information. I would also like to highlight that this app was built by my colleague Ingrid Cheng, and it is based on a published PBPK model, which is cited over here. So the model then gives you an output over here, which is your PK plot and also your trough concentrations, etc. And we aim to build such a model for all different antibodies. This one is only useful for infliximab currently. But thank you so much. And I would like to acknowledge Dr. Shah and Dr. Bees, who's present here, and other MPRINT members, as well as other members of the lab. Thank you.
Thank you. And we are down to our last flash talk presenter, Dr. Hani Faysal, who will be talking about our COPPER project, the collaborative online perinatal and pediatric repository. Hani is a PregMed postdoctoral research fellow with Dr. David Haas at Indiana University.
Hi, thank you for having me today. My name is Hani Faysal. I'm going to talk to you about a project that I hold very dear to my heart, which is COPPER. So First of all, kind of a background introduction to what copper is. Early onset preeclampsia only occurs in 0.5% of pregnancies. So, and it's also a very, very deadly condition, which means that screening for it has to be done early in pregnancy. But being a rare condition, it's very hard to do research on it because of a lack of sample availability. So, how do we get to the bottom of it? There is a gap that we have to. Basically, there is no centralized research tool that could bridge this gap between big biobanks and aspiring researchers. And to address this gap, our objective is to understand the array of biological specimens that are currently being stored from previous research experiments, and to be able to put this into action to better understand maternal and pediatric child health research. Our work is complementary to DASH, and it will help the MPRINT Hub's goal of integrating data and resources across networks, amongst others. So what are our aims? Our first aim is to scout. We want to characterize the landscape of current biobanks and studies that already have previous biological specimens, to kind of see what's out there, to kind of see what kind of research is being done, to better understand the lay of the land. After we scout, we analyze. We go deeper. We see exactly what kind of biospecimens do we have that are related to preeclampsia, so we could connect researchers that are interested in researching about early onset preeclampsia amongst other conditions. And our final aim is to integrate. We want to explore mechanisms for maternal and neonatal pediatric data and biospecimen integration, kind of like creating an interconnecting network between aspiring researchers and already established giant biobanks. So the pilot phase started out, we started out with our MFMU building blocks of pregnancy and Ohio perinatal research repositories. It started out in October 2022. And the aim of the pilot phase was to better understand. We wanted to get some feedback about our survey, and we got suggested changes were the wording of questions, the ability to pause and resume surveys at a later date, and the ability to download a PDF report of the answers. So the current status. Okay. As of today, we have sent out the survey to more than 60 investigators. And we have 32 survey attempts with 15 completed survey that have completed the survey completely from A to Z. We initially started sending out the emails as of October 2022, and we've been trying to send reminders to survey takers that have not completed their attempts yet. And we also attempted to do recruitment. We were in San Francisco at SMFM this February, and we will also be present at the Pediatric American Society Convention in Washington, D.C. These are the status of AIM 1. For AIM 2, we have started to go through the consent forms, kind of understand mainly three main objectives, which is future use, the ability to contact patients for further biosamples, and to see if the DNA analysis of samples if patients have consented to have their genomic and DNA data analyzed. We have already 14, as I was flying in, I got the number 15 actually, and the analysis of the early onset preeclampsia samples will be done by hopefully this June of 2023. And finally, the current status for our third aim, which is the integration, is to review the integration of the biobanks from AIM 1 and AIM 2, and hopefully convene a summer or fall workshop to kind of see the regulatory issues, the consent form issues, amongst others. Thank you so much for your time.
Great. Thank you to Hani and all of our other presenters today. Now our next item on agenda is a short break. We'll take about a 15-minute break and reconvene at 10:15 a.m. Pacific time in our breakout rooms. So for those who gave talks, breakout room 1 is at the far end on the right-hand side, next to the other end of the courtyard out here. And the speakers who will be in there are Zhao, Simone, Zuffa, and Hani Faysal. In room 2, which will be the next one this way, Dustin, Huynh, Sydney Thomas, and Mokshada Kumar. And in room 3, Yaseen, Abbasi, Leeha, Mahmood, and Aamodt. And then we have some faculty already assigned to those rooms. And again, whoever would like to attend those rooms is welcome to. For the virtual speakers, we will have their breakout room in here to be able to give them feedback as well. So we will take a break for now, give you a few minutes to grab refreshments and reconvene. Following the breakout rooms, we will have another presentation back in here at 11. So please return to this room in about an hour. Thank you, everyone.
Much appreciate it. Hi, everyone. I'm Audrey Tremolay. I'm one of the PIs here of the MPRINT grant here at UC San Diego. Thank you so much for joining us for this 3-day meeting. It's a pleasure to be here. I have the pleasure of introducing what I call my colleagues and buddies that really help us spread the word about the work that we're all doing. And it's the communications team from Rady Children's Hospital. I'm here that's affiliated with UCSD. And we have a unique situation with Rady's where it's really the only game in town for taking care of kids. And that puts quite a responsibility on us that are clinicians there on really having really close communication with our community. As a researcher, I will tell you that this team that's next to me has really allowed our community here in San Diego to learn about the work that I do in Kawasaki disease and has taught me how to really provide that information in a way that informs our public without making them scared, frankly. So I thought it would be a wonderful opportunity to share with you guys the expertise of our communications team at Rady Children's, have them come in and share media training as well as social media. And so we're going to have Irena Boostani, Ben Metcalf, and Analisa Jones join us this morning to educate us all on how to better communicate with the public and get our message across. All right, thank you guys. Sure.
Hi, everyone. Thank you so much for having us here today. So I'll start the round of introductions. So my name is Irena Boostani. I am with Rady Children's, and I'm the head of marketing, so VP of strategic marketing and external communications. And my background is 20 plus years of PR experience and crisis communications experience. And I'll kick it off to Ben.
So I'm Ben Metcalf. I've been at Rady Children's for 16 years now in marketing and media relations. Before that, I spent about 15 years in TV news, and my degree is in journalism from the University of Missouri. So I've been doing this for a while. Analisa.
Hi, I'm Analisa. I'm the social media manager at Rady Children's. I have about 6 years of social media marketing experience. And my background is agencies as well as tech, and then more recently now at Rady Children's.
So what to expect from today's session? Part of it is training, and part of it is really just sharing of information and what we're seeing and what we can share in terms of our experience. So one rule for today's session is please do interrupt. So let's make sure that this is an interactive session and not just a presentation that is going to go one way. Chances are you all have a lot of questions, so please either raise your hand, or if whoever is monitoring online comments, raise your hand if we have any comments or questions coming from our virtual audience. All right, Ben, take it away.
All right, so this is going to be more of a high-level overview. Obviously, we can't get to everything with the news media, but just to define some terms, we're talking about earned media. And that's the media that we get for free, basically. It's not paid. And we obtain this sort of coverage through things like news releases, where we send out an announcement, inviting journalists to cover things, whether it's a major announcement, discovery, research, things of that nature. Sometimes we'll have press conferences. We'll make direct pitches to reporters or writers that we happen to know about things. And, you know, one thing, I guess, a warning with this is sometimes you get what you pay for. When it comes to earned media. It's an important distinction to make, that we have no control over what the writers put together. Sometimes we do when it comes to trade publications and things like that, where they want to check their facts. But generally, when you're dealing with mainstream media, you have no opportunity to review what they're writing. Another way that we get coverage is through, this is relatively recent, through expert databases. Reporters can actually ask a question and say, I'm looking for an expert in pediatric orthopedics to speak on this topic or to be interviewed, and that's how they find us. So it can be advantageous for us to have experts sort of in our back pocket that we have available whenever there's questions. And when we're able to place those people in media, it helps raise our reputation as experts in whatever field we're talking about.
So Ben, just wanted to add, so to Ben's point, we don't have control of the messaging a lot of times when we have interviews with earned media. So the other term for earned media is PR. That's what you are probably going to see in your organization, right? So why is PR so popular? Number one reason is because it's the most cost-effective tool in the marketing toolbox, right? So you are just paying for your expertise or your communications team's expertise and nothing else. The second reason is actually credibility, right? So if we are saying something, putting something forward on our social or on our other channels, own channels, it has a certain level of credibility. When it's being repeated by some of the top tier publications, it adds that much more credibility to our thought leadership.
Exactly. And the final one is also relatively recent. Social media. Reporters will find us through what we're putting on whether it's Facebook, Twitter, LinkedIn. They will see something of interest and contact us for a story. So that's also another way to get coverage. Now, let's talk about the media landscape and what it's like for reporters these days. Generally, if you're talking about mainstream general assignment reporters, they have very little time. They're working on, at least in broadcast, a story a day. So they don't have much time to dedicate to what they're working on. They're looking for different ideas. They don't want everyone they interview to say the same thing. When they're writing their story, you can expect them to seek out other voices. Let's say you're an expert on a specific topic, and you say, you know, this is the first time this has ever happened in our state. They're going to make sure that's true. They're going to find another source to verify that, or at least they should, if they're good journalists. So don't expect to be the only one quoted in an article. Oftentimes, reporters are just looking for attribution. They want someone to kind of verify what they're saying. They want an expert voice to put their name next to a quote. One thing we're seeing more of lately is, especially in the online and digital world, are, I guess, I don't know if I would even call them reporters, but people who are trying to build their own brand. They're looking for, I'm sure you've seen these clickbait headlines where it says, you know, 10 foods you should never eat. You'll never believe number 7. And then you click on it. And that's the new reality we're seeing with a lot of these journalists. They're driven by clicks and shares and that sort of thing. Once again, that's for kind of mainstream news. For specific things like research or papers, that's a different animal. And we'll get to that in just a minute. And the final point is journalists are dealing with a lot of distrust, and it's especially relevant. I'm sure you may have seen the Fox News settlement that happened yesterday. So that's definitely in the news. Just a quick overview of the media landscape. At Rady Children's we deal with almost on a daily basis is the local press. And these are the local television news stations, the 4 networks. There's also Univision, where we are next to the border. We do a lot of Spanish language media. There's News Radio, KPBS, and local newspapers and magazines. And this, you know, wherever community you're from, if it's not from San Diego, it's usually the local daily newspaper. There's business news, regional magazines, and also hyper-local news outlets, which are a good way to get specific stories placed in very distinct communities. And here we have these small publications like La Jolla Light, Rancho Santa Fe Review, places like that. But the local press is interested in things that are regionally interesting, that you can somehow apply to this area. If it's not interesting to folks in San Diego, they're probably not going to cover it. And human interest stories are also a big draw. You know, we work with TV news a lot, and anytime you have a patient story, it's key. You can have the most fascinating research or findings, but if you're not able to tie a human face to it or a patient, then it's going to get a lot less notice. So, when you're looking for coverage of something, always be thinking about the patient component and how it has a personal impact. So within the media landscape or broadcast news, and just based on this medium, it's visual stories they're interested in. Once again, talking heads and sort of papers that are hard to digest aren't a great fit for broadcast news. We're fortunate at Rady Children's where we do a lot of patient stories with kids, and that's great for broadcast news because you've got kids and you've got happy endings and great stories and that sort of thing. But generally broadcast media, they like celebrity news, dynamic speakers. They're fast. One day you hear about the 24-hour news cycle. It's very true when it comes to broadcast. And the stories have to be simple. I think of a quote that really resonates is a reporter's knowledge is a mile wide, but an inch deep. So they know a lot about a lot of things, but not in great depth.
And to that point, if you have a story that is very nuanced, broadcast would probably be the last place you would try to pitch it. So most of the time you're going to go to very credible online and print publications first, and probably stay away from broadcast because it's just going to be snippets.
Right. So where you want to look is the trades and journals. And these are specific to some examples here: cardiology, spine review. I threw in the maternal fetal and neonatal medicine journal for this group, might be interesting. But these are where you are able to find an audience for research and for papers and for studies and things of that nature. New trends. I spent some time at the Genomics Institute at Rady Children's for a bit, and there's a whole subset of media that just cover genomics. Genome Web is one of them, and they will generally have more time to dedicate to the story. They're not like the one-day turnaround that we talk about with TV news generally. So this is kind of a sweet spot when it comes to talking about things that's very specific. They can be a little bit more technical. And you'll have reporters who have some experience. A lot of them are PhDs, so they have a better, deeper well of knowledge in that specific area. So to me, this is what you would want to target if you have very specific things you want to talk about that aren't necessarily of interest to just the wide generic audience. And now we're up to you, Irena.
All right, so let's talk about the interviews.
Yes.
There are a couple of ways that the interviews are going to come your way. One is your PR team is going to proactively pitch an idea to a reporter, something that your organization wants to talk about. That's one set. And then the second set is inquiries from reporters to your organization, and you are being put forward as the expert. So it is the second subset of interviews that you are going to scrutinize the most. Why is the reporter interested in this particular subject? And this is what gets us into the conversation of tough interviews. When you have the easy interviews, and the easy interviews, we call them the fluffy ones, right? Is what's the best part about your job? And tell us about your most interesting cases. And these are the softball questions that when you read the article that comes out after that interview, it's very fulfilling, right? So the second subset can be a little bit tricky. So we don't know where the reporter is particularly coming from. So since you don't know where the reporter is coming from, chances are your PR team is going to vet them. They're going to vet the publication, and they're going to vet the reporter. They're going to look at what the reporter's beat is. They're going to look at how this reporter covers their beat, and any kind of red flags that might come with the previous articles. But it is really good for you even if your PR team has done all the background work, it's really good practice to look at their previous article regardless, because you might actually, you might actually pick up on something maybe your PR team is not going to be able to pick up by virtue of clinical knowledge. So it will also help you figure out what their point of view is. The next part that you want to work on is what are you going to be your key messages that you want to deliver. So let's say you decided we are going to move forward with this interview. The reporter is asking about a particular trend that they're seeing across the region or across the nation that is specific to your specialty. Then you want to work out what your key messages are. Typically, you don't want to have more than 3 to 4. And have them as sound bites, no longer sentences, because it's much harder to remember. They oftentimes will not come across as very authentic. Just keep them pretty short. And I'll tell you how they're going to help you in those interviews. So actually, let's talk about the types of interviews that you might have based on the type of outlet. So the first one, the hospital or research news, these are feature articles. So for example, your organization put forward a big piece of research that is really has breakthrough type of results. The reporter comes in, your PR team, first of all, pitches that out. The reporter comes in and has a lot of questions for you on what this means for the region, for the nation, for the specialty, for the health, for the patient population, and so forth. So that type of an interview is going to take a little while. So the reporter is going to try to dig a little bit deeper to understand what it is that you're trying to do. The second type of interview is kind of similar to that. Also trying to understand kind of the underlying information in the background for the type of story. Also a little bit of a softer tone, but oftentimes it's going to be driven by a very large trend that is typically on the national level. That can be coming from your trade publications, so talking about your own specialty, or it can come from national publications. A lot of times this is going to be USA Today, it's going to be Fortune, CNN, and so on and so forth. And they're trying to dig deep into what is happening across the nation with, let's say, a pediatric population. Quick turn, very, very quick interview. On average, it lasts about 10 to 15 minutes. Also some sort of a trend, but we know this trend is not going to last very long. So in our experience, it's the triple virus. Perfect example. October, November, I think this is all we talked about, is the triple pandemic throughout the nation, right? That was exacerbated in our ERs by mental health crisis. This is all we talked about. Most of the interviews were those quick turn interviews. There was one from Fortune that turned into a feature interview where the reporter was trying to understand how that dynamic is actually affecting healthcare organizations across the nation between the viruses and mental health, and how organizations were dealing with those. And then broadcast, to Ben's point, those are usually 5 minutes or under. And this is where your sound bites are going to be the most helpful, because you are going to be cut. Right? So you want to make sure that you communicate your key messages as fast and early in the interview as you can, and most likely repeat them, because this is where things are getting cut quite a bit. To Ben's point, yes, there are factors outside of your control. Let's talk about editing. This is going back to the message points. Try to get them into the interview as fast as you can. And also be careful about what else that you are saying. I'll give you an example. We had an interview, Healthline, talking about the effect of education right after the pandemic and masking on the kids' mental health. It was a great interview, very balanced. Our psychologist was interviewed. And obviously, we all worked on the messaging together. We know what we are saying. The headline comes up, and masks are hurting children's mental health. Right? So did we say something like this? No, we did not. Was there potentially an implication that some kids might have felt uncomfortable? Possibly, but that was the result of this. So this is why the background information on all of those reporters is so critical, right? I had another example of another CEO from tech company who was talking about that he came on board of this tech company to revive the company, and the headline read, the CEO does not want the company to die. It was very, very dramatic. So earlier in the presentation, Ben was talking about, the click rates. Unfortunately, we are seeing a whole lot more of the reporters, a lot from digital journalism area that are trying to build up their own reputation. And unfortunately, the clicks is the way that they prove success. So we need to be very careful about that. The second part of the expectations is don't be upset if you're not a huge part of the story. That is absolutely normal. We also recently had an interview with Politico on mental health and funding for mental health on the Hill. Ended up with probably a 20-minute interview, one sentence at the end. But it was probably one of the most powerful sentences that we've ever had, because we all know that Politico has a very large following and it has the right audience for that type of a conversation. So it's a huge success to have one sentence in an article.
And just to follow up on that, it's also important for the future, because other journalists will see that article and realize that your expert was quoted in a large publication, and that's sort of, down the road you'll get more publicity or visibility on that specific expert just because of that one sentence in the article.
That's a great point. A lot of times journalists, especially from top tier publications, the first thing that they're going to do is to do a little bit of a background check on you as the expert and say, where else have you been quoted? So that is really important to keep in mind. The first bullet point, I think, is really the most critical one if you are faced with a tough interview. And sometimes you might actually not know that you are faced with a tough interview. Another example, we're having a specialty focused interview, and all of a sudden the reporter asks, so what do you think about California regulation on X, Y, and Z? Right? So we didn't know we were coming into a tough interview, but there it was. So the first things first, a question is simply an invitation to speak. It doesn't mean that you have to answer that question. So your typical answer is going to be, if you're a clinician and you're not familiar with California regulations, well, I'm not an expert on California regulations, but I can speak to you on neonatology and bring it back full circle. You might actually find yourself using that sentence more times than you expect to. The other way to bring this around is I would be happy to put you in touch with an expert on X, Y, and Z, but I'd really love to cover the topic that we were talking about. Allowing the reporter to finish their question or comment. Sometimes reporters can ramble a little bit and the question can get lost. In that case, please ask the reporter. So first of all, let them finish their question. But if there is something that is unclear, please ask them to restate the questions. It can be that they're just really, really busy, or it can be sometimes done on purpose. So clear questions are always a must. And then the next one is evaluate the question. Could this potentially be a loaded question, right? So there are some, and I think we're going to talk about this a little bit more, but could the question be, so how bad do you think California regulation is on X, Y, and Z, right? So evaluate what is it that the reporter is asking. And I think taking a second to formulate your response is going to serve you a ton of good. Sometimes people feel like as soon as they ask a question, they need to be jumping in. It is much more prudent to take a second. It might feel uncomfortable for you as a long pause, but if you actually saw yourself on video, it doesn't look uncomfortable at all. To the other person, it actually looks like you're just thinking. So it's much better to take a pause and just formulate your answer, because once it's out there, it's out there. So we just want to be very, very thoughtful.
And just one more thing on that. It's actually a tactic that some reporters use is silence. You'll feel the human nature is to fill the space, because you're just sitting there in silence and they're just kind of waiting for you to say something. So if you can be comfortable with silence, it's a good thing.
Agree. Going back to your key messages. That is, those messages are going to be aligned with your organization and your constituents. And these are going to be typically your patients, your employees of the organization, partners, community. In our case, for example, it's California regulations as well, right? So we have to think about, in this particular case, right? So do we want to comment on California regulation from a clinician standpoint? Probably not. I think we talked a little bit about going kind of straight into the key messages when answering the reporters' questions. And then actually this last point is, a lot of times reporters might get a little bit lost in the clinical language. We actually find ourselves in that position sometimes too, and we say, please, can you repeat that, but a little bit in layman's terms, so that typically if they're asking you to repeat something but explained, it's because of their audience. It's if they're writing for the general mainstream population and they couldn't quite understand what you're saying from the clinical standpoint, they might ask you to restate your answer. So typically that's what they would be asking is, how do I explain that to general mainstream population?
Right, and you think about USA Today, they're notorious or well known for writing at a 6th grade level.
Mm-hmm.
And sometimes when you're dealing with mainstream media, you just keep that in mind, that you really need to keep it simple. That's different for the trade publications where you may be talking to a PhD or someone who understands the topic, but generally keep it as simple as possible.
Off the record, there is no such thing. But there is such a thing as on-background interview. This only happens typically with top-tier publications, the Wall Street Journals and the Fortunes of the world, if they are planning to cover a really, really big issue. And what they might do is go to different stakeholders and have several on-background interviews, and they will state this is on-background interview. I still wouldn't say anything that is super controversial or would get your organization, in hot water. But there is such a thing, and typically it would take them a month or two to write that piece. So you would know that this is probably a very large trend that they're trying to cover. But when you're speaking in an interview and the reporter says, off the record, what do you think about your competitor? That's another thing that we're going to talk about, is talking about competition. There is no such thing. Okay, so that regardless of the publication, everything is on the record. No comment, not a preferred method of response, right? So you have to, in that moment, think about why is it that you don't want to answer that question, right? So am I the right person to answer this? That could be your answer. I'm probably not the right person to answer that question, or I'm not an expert on XYZ, right? And go with that. But I can tell you about the other things that we've talked about. So that actually speaks to the next point, right? So if you don't know the answer, don't try to answer it, because a lot of times we would see, someone starting to answer it, but if you actually don't know, if you're not a subject matter expert on that, it becomes a little bit of rambling. So make sure that the appropriate experts are involved in this. When faced with a leading, rambling, confusing question, restate the question back to the reporter. This sometimes can be used as a tactic to get you to say something that you actually don't want to say out loud. And then in any case that the question is loaded, so in case of, how bad do you think California regulation is on XYZ? We had another one, and that was probably closer to you, is pediatric formula. Is we got a question from a reporter. How do you think the organizations that produce XYZ formula should be reprimanded about? And it's like, that is not something that's, we wouldn't say something like this, that's how, those organizations should be reprimanded. So if you're faced with a question that implies your statement, correct that as soon as possible. This is not what we stand for, or this is not what I'm saying, or this is incorrect. So this is probably the most direct way, is like, this is incorrect, and then please correct it. deliberate provocation by a reporter, unfortunately, it does happen. This is going back to Ben's point about clickbait headlines. If they get you to say something controversial, oftentimes it's going to make it into the article. Most of the time it's going to make it into the headline. And has it happened? 100% it has. So you just need to be careful about that. And then insider anecdotes and competition. That is a big no-no in our world. Is do not comment on your colleagues, do not comment on your competition. And that is, if you are asked a question, what do you think about Stanford's pediatric cardiology care? We do not comment on our, on our colleagues in other hospitals, but what I can tell you about and bring it back to your own messages. So that is the second part of why your key messages are going to help you, is if you stick to your key messages that you've outlined in the beginning, that is all the reporter is going to be able to cover. And that's at the end of the day, that's what you want, is making sure that your audience, and in this case, your audience is not the reporter, your audience is who the reporter writes for, sees the key messages that you and your organization have worked out. Patient privacy, I don't think we need to explain this to you, how important that is. And then, of course, any financial information, feel free to use PR people as the punting down the road. Would be happy to put you in touch with our PR team to send you the follow-up items, but definitely do not comment on the revenues and the financial projections of your organization.
So the next section is social media, but I thought we'd pause here if we have any questions about the news media. Yeah.
So, Irena, you use the word "we" a lot, talking about communications team and the person being interviewed, but you didn't address when the person being interviewed. You use the term "we" a lot, which I really appreciate, but you didn't address when the person being interviewed was cold called by a reporter and it didn't come through the communication team. So can you comment to folks here about how that should be handled?
Absolutely. That's when your PR team comes in again. And your response to the reporters, I would love to put you in touch with our PR team so they can coordinate. So the PR team is going to get in touch with the reporter, but the first thing before they coordinate, what they're going to do is the background check on what this reporter is interested in, what their articles look like, are they controversial in any way, and so on and so forth. And then together you can decide if you want to take that interview or not.
Yeah, I think the takeaway is no matter, how simple it sounds, or you always want to contact your PR team, even if it seems completely benign, just let us know and we'll coordinate it.
Because at the end of the day, you don't know, the reporter might be focused on one thing. Even in the interview, a thought might cross their mind and they will ask a question that that might stump us. So we would just probably do a little bit of due diligence of what other topics they might be covering.
And when we do interviews, almost always a PR person is on the call or the Zoom with you to help facilitate any issues that might come up.
Thank you for that, and I appreciate.
Everybody hear me okay? Perfect. I appreciate that, especially since I early on did not do that and found myself in hot water in the sun times, and I regret that deeply. So I always try to give that advice as well. So thank you.
Share with us, what kind of questions were you asked that you felt, or approximately?
So this was related to the death of John Travolta's son, who had incidentally Kawasaki disease, but also had a number of other issues and passed away not due to KD. And I was young and excited to be interviewed and got a cold call from paper. And I'd been doing a bunch of other interviews related to that same topic. And I thought, oh, this is great. I'll just answer. I'll get on the call. And I called, and what got published had nothing to do with anything related to Kawasaki disease. And I was in this quoted. It was just really a sad example of what can happen in a moment of enthusiasm. So, you always have to filter it through your PR people. Basically, that's the take home message. And I have been called before and it seems like they're in a rush, and not my problem. Call our PR people. If you're in a rush, you'll adjust your deadline if I'm that important to you. Right. So I think that that's another thing, is like, just put it through UCSD or Rady Children's Media if you're here in the San Diego area. The other question I wanted just to ask for is that a lot of the work that we're doing as part of this network and other networks is very research based. And so it's the question of the hook. And when you've got a patient facing situation, that hook can be pretty obvious. But I wonder more for science research, how you try to find that hook and how to have that interaction so that you guys know that if you're looking for someone in a particular area, how to build that bridge to something that will become a hook, if even like it's interesting to us, but it may not be that interesting to the public. How do we make that bridge?
Sometimes it's going to be possible. Sometimes it's going to be tough, right? So the first thing that we assess is how big of a, let's say, population this type of news is going to affect. And the smaller the population, the harder it's going to build that bridge, right? But generally after that, the question becomes is this research part of best of, new type of outcome, first in the, right? So those are becoming the hooks. Are we the first in California, or are we the first in the nation? Is this information so new that nobody has ever talked about this, right? So those become the news hooks, whether it's scientific information or the mainstream information. For mainstream, for top tier media, typically it's the larger the population that is going to be affected by this news, then the higher the chance that we're going to be able to place this article.
I'd also add, you can always brainstorm with the media team, because we come from the perspective of generally, the average consumer of news, to come up with that hook. I think making an analogy for your research to something that people will understand is a great way to do it. If you look at the Salk Institute, a lot of their press releases they do are really smart that way. They'll make an analogy to something that the average person will understand that you may not have thought of, but we can work with you to sort of brainstorm that hook. And I think we also know what journalists are interested in. So. We can move on to social media.
Yes.
Yeah.
Sounds like.
Oh, online question.
Okay. So there's a tip from Phil Wilson, who's online. He was given that I found to be a tip I was given that I found to be useful is to not waste the mic check moment instead of about 1 to 2, 1, 2, 3, and state your main point during that time.
Very cool. Interesting. Actually, that brings up another tip. If at the end of the interview, the reporter asks you, do you have anything else to add? Please restate all of the messages that you have worked out before you went into that interview. That once again will ensure that your message comes through. Yeah. To Ben's point, we don't have the guarantees, but it increases the chances.
Yeah, the answer to the question is always yes, I do have something else to say. And even if you've said it 2 or 3 times before, it's an opportunity to restate it one more time. And that sort of conveys to the reporter that this is the important takeaway.
All right, so moving on to social media. So we're going to talk about social media that you're using on your personal channels and how to kind of leverage your own audience to share your research, or maybe if you get some sort of coverage through your PR team, you can share it there. Just how to be smart using your personal social media channels in promoting content related to your organization. And social media, it's the audience is huge. There's billions of people who are on social media around the world. So there's a lot of opportunity there to connect and share information. But when you're doing it for your organization and talking about your work, there's a little bit more strategy that has to go into it than what you're doing for your personal use. So that's just what we're going to cover right now. So basically, if you want to start, promoting yourself and your work on your social media channels, the first question that you want to ask. Is that better? Okay. So the first question that you want to ask yourself is, what is your goal? You don't want to start posting until you answer this question because it's going to determine basically how you're going to approach the rest of your social media and posting. So is it to expand your network? Is it to share your research findings? Is it to reach patients? All of that is going to determine where you're going to post and how you're going to post. So once you have your goal, that's how you'll define your audience. You'll choose a platform, you'll develop your voice, and then continue community engagement. So providing that valuable content so that people come to your channel to get information. So to start, we'll cover the primary social media channels. I'm sure you are all familiar with most, if not all of these channels, but we're just going to go over briefly the different audiences and the different content that performs well on all of these channels. So LinkedIn, these are the professionals, as you probably know, and on this platform, your research results, articles that you've written or that maybe you're featured in, any blog posts, media coverage, this all performs super well on this channel. And you do want to add, that visual element to it. So pairing it with a video, a photo, graphics, that all will help increase engagement when you share on LinkedIn. Instagram, this is more of the parent audience. There is obviously younger people on this as well, but just wanted to note that, almost 32% of the platform is ages 25 to 34. And on this channel, it's a lot more visual. So think photos, videos, Instagram Reels, those videos perform super well. There's also story content that you can have up for 24 hours. And on this platform, you can share behind the scenes of your research or, things that you're doing with your team. You could also, engage with the community, like any sort of work that you're doing in the community that maybe it's kind of coincides with what you're doing in work. It's a great place to promote it. As well as just timely content. There's a lot of trends on Instagram. You can speak about trends on Instagram. So that's a great place for that. Facebook, so 36% of users on this platform are 45 years or older. So I think grandparents and parents, I know for us, a lot of like educational, informational type content performs super well on this platform just to kind of spread the word about different things. But again, videos, photos, again, perform very well on here. There's also story content, and I've been testing it more for Rady Children's, and they definitely get seen and get interaction. So another good way to get out quick, timely content. And then Twitter, really anyone can be on Twitter. We've talked about the media. I know for Rady Children's, our Twitter, the media has picked up things on our Twitter account. But it could be parents, grandparents, professionals, really anybody can be on Twitter. It's a wide audience. And you can provide really timely updates, news, and information on this platform. That's the best place to put that. And then TikTok. So if you're trying to reach the more patient audience in pediatrics, this is a great place to be. Just wanted to note that 25% of US users are ages 10 to 19, and nearly 23% are ages 20 to 29. So that's a big audience. It's exclusively video content. It's really based on trends. There's sounds that you can participate in, and your videos and content will get seen more if you're participating in those sounds. And the content on here is a lot more authentic and relatable. That's the content that performs well. So it's a little bit more personable as well.
And Lisa, if you want to spend just a little bit of time, so Twitter, what we are seeing on our platform is physicians posting the conferences, for example, or events that they're participating in. And it looks like physicians are looking for other physicians to see which conferences that you guys go to. The other use is sports partnerships. A lot of sports updates. If your organization has any types of sports partnerships, it's going to be usually Twitter. And then TikTok, for us personally, we had lengthy discussions about if we are going to support TikTok as a platform. We decided not to, for a lot of obvious reasons, but the two for us that stood out were obviously concerns, very serious concerns about data, but also very inappropriate content that is targeted towards children.
Yeah, and TikTok, they don't do the best job with, regulating the age requirement on TikTok. So there's a lot of child exploitation, unfortunately. And so for us, it's a no-go.
So for us, as our organization goes, LinkedIn, Instagram, and Facebook are the platforms that we support quite a bit. And YouTube is our growth platform.
And then just a note on Doximity, I'm going to pass it over to Irena, but many of you are probably already familiar with Doximity. Your organizations might already be leveraging this. But, yeah.
So Doximity is super interesting to us as marketers. And part of it is because it's a little bit of science and a little bit of mystery. Doximity has several parts to it, right? So it's the physician network. And from that standpoint, this is why we included it here. It is a network for physicians. So it doubles up as a proxy, as a social media proxy for physicians. The other part of it is actually a marketer's tool, and we work very closely with Doximity. Do those two parts connect organically? Not always, but there is a tremendous opportunity to connect those. So we are working with our physicians to connect those two, what it is that they're seeing from their perspective, because we actually cannot see as marketers the physician's view. And our physicians don't always see everything that we do on the marketing side. So for example, this is a great tool to Analisa's first slide. What's the goal for you to be on social? If the goal is to increase the network, Doximity is a great tool for that. And it looks like you have organic opportunities to expand your network on Doximity, but there's also the marketing opportunity, the paid opportunity of colleague connects as well. So do work closely with your marketing and communications team if you want to create more of an organic presence on Doximity.
All right, some just quick social media do's and don'ts if you do start posting about your work or anything related to your organization. So regularly post content, quality content on the platforms that you choose. The more consistent you are, the more you'll grow an audience and the more engagement you'll get over time. And with that, post original content, or if you are posting third-party content, make sure you give proper attribution to whatever platform it is. I know for us, whenever I post a coverage article or something, I always give like Fortune a tag just to make sure that we're giving everybody credit. Use imagery or video to increase engagement. I think we listed that on all of the platform overviews. The more videos or photos or visual aspect of it, the more you include that, the more engagement you'll get. Reply to comments within 24 hours. This is just best practice. If you're growing a following, you're going to inevitably get more comments, and people expect pretty quick responses on social media. So just as a best practice, respond to those. The next one, remove inappropriate comments. Again, as you get a bigger following, you might start to see more comments from like trolls or just, people who are maybe don't agree with you. And if it starts to escalate, please flag that to your corporate social media team. So your organization's social media team or marketing team, and just let them know, because they can help you respond effectively or not respond. They'll guide you in the right direction. So definitely do that. Don't use explicit language or images, reference alcohol, drugs, weapons, or illicit content through words or images. This is just something that we require on our end as well at Rady Children's. And don't communicate with your current or potential patients regarding their care. This is a big one. Please funnel anything like that to the appropriate channels at your organization. And then please don't also dispense medical advice. This can get hairy pretty quickly as well.
I think all of you in the audience know this, right? So we shouldn't even be talking about this. Unfortunately, we actually have seen instances, and we've done a lot of due diligence of how medical professionals look on social media. And we have come across, thank goodness, not in our own organization, but we have come across medical professionals who cross that line into actual medical advice. And it was actually on TikTok as well. And so therefore, if you start looking at the rise of physician as an influencer articles, you'll find quite a bit, right? And usually it's TikTok and Instagram that are going to be the two main platforms for this. It is out there. So this is why we are putting it in our PowerPoint slide, even though it's self-explanatory.
All right, so some considerations. Again, before you even start posting, these are just a couple of things to keep in mind. Anything that you post is a reflection of your reputation and your organization's reputation. So just always have that in mind before you post. Even if you don't mention your organization, people can easily look you up online and see where you work. So even if it's not included, people can easily access that information. So just keeping that in mind. And just as Irena said, physicians as influencers, we're seeing that more and more, and we're seeing some physicians doing a really great job being very thoughtful with the content they're putting out. And then, others crossing some lines. So again, just keep that in mind. You can always include a disclaimer in your bio, which we've included here, just saying that, you know, the postings, the thoughts and opinions are your own and not of your organizations. That's another way to make sure that any lines aren't crossed there. We also recommend, you know, don't identify as an employee, omit any branding. So for our physicians, don't include any Rady Children's branding, and you know, try not to take photos or videos in like really identifiable locations if you are, you know, putting out your own content and opinions on your social media. Anything to add there?
One thing that I'd like to add, some of the asks that we have from our own organizations, can we start our own Instagram account for our subspecialty? And sometimes it's the subspecialty, right? And there is a reason for it. Let's say we want to spotlight our culture, or we want to focus on recruitment. The questions that you want to ask yourself if you are in that position is someone going to be able to manage that account from start to finish and very consistently? Because we've done our due diligence and we've looked at, okay, so how do other hospitals and other organizations manage this? How do those other accounts look like? And for the most part, what we found is 100 to 200 followers and kind of haphazard posting, right? So it doesn't grow. So any time that you want to start a particular account for a subspecialty, let's say, you want to think into the future and how you're going to continue to grow that audience. And the way to grow that audience is consistent posts and diversified content. That takes time. So sometimes you might say, you know what, we have the staff, we have the commitment to do this on our own, and that's great. And other times you might say to your social media team, we actually don't have the resources to manage this yourself, but can you help us out, help us with that goal, whether it's profiling our culture or with the goal of recruitment? And we've kind of found it to be able to work both ways, but treat it on a case-by-case basis.
Yeah, and to your point, I mean, speaking as the social media person at Rady, I'm always looking for content. So I'm always willing to partner with other teams if they want to start their own account. I'm like, please give me that content, I'll promote it for you. And to a bigger audience, because you have to build a network if you're starting your own. And then we already talked about, you know, TikTok, and just a last note here, a lot of those TikTok trends do make their way onto Instagram, and Instagram is, you know, a trusted social media platform. So you can definitely do very similar content, but on Instagram, and reach a very similar audience. There's still a lot of that younger audience on Instagram as well. And I think that's it, unless there's any questions around social media.
Actually have one. So you talked about using stories. Can you discuss when you would use a story versus a post on Facebook or Instagram?
Yeah, so I think that goes back to, at least at Rady Children's, we get a lot of requests, and some of them make sense to like live on forever on our feed, and some are more of a quick thing. So I think a good example we could use now is April is Child Abuse Prevention Month. So we did, you know, a kickoff post with our doctor who specializes in that. But throughout the month, every Monday, we've had her, we took talking head videos of her giving tips on how to prevent child abuse and neglect. And that's performed super well. We've gotten a lot of great responses, but again, we weren't going to post or be able to post that frequently on our main feeds. So having it live on the stories periodically throughout the month has been a great way to share that. Yeah.
And the second one is the Reels too on Instagram. So the Reels we use quite a bit, especially for culture and patient stories. And those are actually pushed out to the audiences outside of who follows you. And that's a really great tool to raise awareness, and they will just populate in your feed.
Yeah, just Instagram favors that right now. So it'll push it out to more people. So that's how we've been getting a lot of new followers.
Hi. So basically, I had a question because we were talking about social media, and it reminded me of a physician back home. Basically, she was a sexual health medicine doctor, and she would always get shadow banned on Instagram and on TikTok, and she would get her account deleted. So when you get to that point where the content that you provide can be twisted around, can be misinterpreted, can be sometimes even used against you, would you persevere? Would you start using certain buzzwords as to not get shadow banned? Would you drop the social media topic completely, or is there a way to push through these adversities that you would encounter when you promote content that might be sensitive or that might be a bit raw to a larger audience?
Yeah, that's a really good question. And we have to vet everything that we post for that reason too, because we don't want that to happen. I think a good example is actually, we had a patient story of a kiddo who had a heart transplant, and he asked his doctors after if he could hold his heart, his old heart, after his transplant. So we were in communication with the mom, getting the patient story, and they really loved these photos of the child with his heart. But
you mean the 3D printed heart?
No,
Right?
his actual.
Oh, his actual heart?
His actual
okay.
Old heart. So we vetted it, and we're like, if we post this, we could get banned or get flagged as inappropriate content. And so for that, we actually worked with our cardio team to be like, hey, do you think this is appropriate to post? And it's a reflection of your team, of course. And, after talking with him, we decided, let's go with a photo without his heart, of him just like listening to his new heart. And so I think there's ways that we can pivot so we can still get the message across. But again, like, work with your marketing team and they can help you pivot appropriately to kind of avoid that.
I would also probably look at other platforms because Instagram is going to continue banning that content, right?
Yeah.
So there is no way around it. So I would possibly use the social media as a tool to point them to another platform. So blog posts could be, right? So if your colleague has, their own website, and use the blog post as all of the content that they want to put there, but then use the social media as the conductor to take them to that blog post. Because the social media is most of the platforms are still going to ban it since it's explicit.
Yeah. Does that answer your question? Okay. It's a tough one.
All right.
I don't see that there are any other questions. Thank
Great.
you guys for a great talk.
Of course.
Yes.
Thank you for having us.
So next on our agenda is our networking and mentoring lunch. So for those of you online, we will join back approximately 1 p.m. Pacific time. So about, I think, an hour and a half, or 20 minutes. Nope, just an hour. So thank you for joining us. For those who are here, we have lunch outside. There's a pasta station and salad available. And then we'll be eating in here. As you know, we've kind of assigned tables. I know there's some missing people, so we can join tables so that you can get the mentoring aspect. The goal is so that our trainees and early career investigators will have the opportunity to talk with some and get career advice from more experienced investigators and attendees. So thank you, and we will see you back here at 1.
So next, let's bring it back together. Let's bring it back together. So how are you feeling today? Are you green, yellow, red? Are you feeling engaged in your work? Maybe not so, or a little bit. There's some interesting research coming from an app called Kona, and it's implemented into different kinds of technology, such as Slack. And it's a conversation starter. It's a way of working within your team to see how they're feeling today as a check-in, similar to what we started with today. Today, I'm going to talk about the role of servant leadership and overcoming burnout. I have no relationships of any kind with a company whose products or services in any way relate to the practice of medicine, medical education, or research. Our agenda, we're going to start with some connection and well-being exercises. I started before we began by checking in with who is at your table, or who is online, or who's in the room with you. So often we go into meetings, we go into presentations, we're carrying a lot of stuff. We're looking for tools and resources, the language to be able to communicate with one another, to be able to say, you know, I'm tired today, or I'm just not feeling good, or I brought with me a concern from home. How can we present ourselves in a professional way, but in a way that makes people feel like we're human? I'm going to talk about my impetus to act. How did I get involved? How did I come here today? I can tell you it started 13 years ago. I'm going to talk a little bit about the underlying cause to burnout. What are the conditions that lead people and even complete industries to high rates of burnout? We're then going to talk about how we identify our organizational culture. What is an organizational culture, and what is our individual culture, both organizationally, societally, and industry-wide? And then I'm going to introduce and talk a little bit more about the transformative opportunities by cultivating a servant-led mindset. Before we begin, I'm going to invite you to the 4 breaths presencing practice. So I teach at a university right now where 80% of my students are military. When I first began to adopt this in every single class and in my presentations, I'll tell a story about someone named Tony. He'd been a Marine for 30 years. He was the size of Hulk. He was just huge, tough as can be. And he said, I don't do that. I said, well, I'm the professor. You're going to do that. And so, all right. Well, by the time I had him for 6, 7, 8 classes, I would put up this slide. And he would be the one to lead the class. He would be the one to immediately go into mindful meditation. So I invite you, take a moment and close your eyes. Sit upright in your seat. Place your feet flat on the floor. Imagine roots growing into the floor or to the ground. There we go. From your head, imagine a string tied to the sky. Our first breath is for ourselves. Inhale from all directions, like a balloon, all that we are most in need of for the thriving and well-being of ourselves. Purify that intention in our hearts, and exhale all healing, renewal, and wholeness that we are most in need of. Our second breath is for our family, whatever constitutes family to you. Inhale from all directions, all that we are most in need of for the thriving and well-being of first for ourselves, and now expand it to our families. Purify the intention in our hearts. Exhale all healing, renewal, and wholeness that we need in all directions. Our third breath is for the well-being of our community. Inhale in all directions, all that we are most in need of for the well-being of ourselves. Expand it to our understanding of family, and now include our community, which is our organizations and our universities. Purify the intention in our hearts, and exhale all healing, renewal, and wholeness that we need in all directions. Our fourth breath is for the well-being of the world as we know it. This is who we've known, have known, or yet to know. even those who may not like us. Inhale from all directions, all that we are most in need of for the thriving and well-being of ourselves, our understanding of family, our community, and now expand it to the globe. Those are the communities that we serve, the people in those communities. Purify the intention in our hearts. And exhale all healing, renewal, and wholeness that we need in all directions. When you are ready, wiggle your toes and come back into the room. So my impetus to act. 14 years ago, I was deeply invested in my career. I loved the career that I had built in the insurance industry. I had an opportunity to work in corporate management as a marketing director, as a regional recruiter, trainer, and developer. And then I decided, you know, as I talked to those agents that I was cultivating and building and training, they kept asking me, what credibility do you have if you've not been an agent yourself? Well, I was invited to start my own agency. And so for 3 years, I built an agency from scratch, from zero to $850,000 a year. But I moved to a different district, and that different district had a district manager with a terrible reputation. He'd been with the company 46 years. He'd always managed to just escape getting in trouble. We know that one. We all know who in our organization tends to just slip by. Meanwhile, the company that I worked for was slowly shifting the attention away from the agent to the all 1-800 number, the call center. And so over the period of the 3 years that I was an agent, I began to see that shift in trust away from the expert to the 1-800 call center. And that increasingly diminished the role that I played with my clients. In my 8 years in the insurance industry, I began to see that my value no longer mattered to the company. I became a cog in a wheel. Meanwhile, this district manager began to shift the contract because in the 72-page contract, there was small print that said, contract can change at any time. So I was set up for a case of burnout as a result of toxic stress. Overlapping all this, when I think about this experience, my husband and I discovered that we were expecting our first child. We were so excited. He was an older father. He was 43. I was 31. And we couldn't wait to be parents. During that time, my contract changed. When my district manager learned that I was pregnant, my contract substantially changed. We hear these stories day in and day out. And the toxic stress of that experience led to my son's prematurity. I had a lot of complications in my pregnancy. On December 8th of 2009, a profound experience happened. I had moved from the San Francisco Bay Area, world-class healthcare, to a rural community in Southern Oregon. And I had approached my obstetrician a few times. I said, you know, I'm not feeling listened to. The stress is extreme. Oh, but the data says stress is not a high hazard for prematurity. We now know that's not true. But at the time, that's not what the literature said. And I had also begun to show signs of preeclampsia. We can imagine where this is going. On December 8th of 2009, a huge blizzard came in. Huge. It was a 100-year blizzard. And I went to the hospital for a hand reconstructive surgery because I had fractured my hand. Coming out of the surgery, the nurse said, brought my 155 or 115 blood pressure to the attention of the attending nurse. And the nurse said, just go ahead, discharge her. Discharge her, it doesn't matter. Just, we have numbers to keep. The nurse, brand new nurse, said, discharged me and recommended I go to the birthing center. That was a very smart move, because that night, the helicopter was grounded. The pass over the 5,500-foot mountain was closed. I was transported by ambulance to a fixed-wing plane over a 5,500-foot mountain, and I landed at a hospital that was very different. It was the tale of two hospitals. And as I got pushed into the birthing ICU unit, I saw a very different personality between. I remember being at such incredible peace. The terror, the fear, the panic that my life was at stake at this hospital was suddenly, I'm watching, it almost felt like a nutcracker. And I described it on the front page of a newspaper a few weeks after that. It was like the nutcracker. Every single physician knew their role. They knew what to do. And in the back of my mind, I said, okay, there's something that they're doing at this hospital that they're not doing over here. There's something different between these two. So we spent 56 days in the NICU, and the years of being beaten down and feeling like I was inept in my career. Suddenly I was being trained through the hand-over-hand nurse care. I was learning how to care for my son. They taught me skills that I still use today in academics by practicing listening, by listening to and connecting with people. These were life skills. So I thank our nurses for teaching the basics of what it meant to first lead a family, to lead myself, to lead a family, to lead my son, and be able to train others on how to care for our son. Fast forward a couple more years, and I knew that this meant something. I was meant to do something between these experiences. At the time, there was a convergence of a couple different experiences. The role of the patient leader, the NICU parent leader, was emerging as an important role to collaborate with healthcare systems. But how do we determine our credibility? What is our credibility? How do we go beyond telling our story and actually contribute in meaningful, constructive ways? What do we need to do? And so I had a couple more years to stay at home with our son, and I had an opportunity to pursue my master's in organizational leadership with a concentration in servant leadership. I began to see how my experience is a very familiar experience. No longer that I could do something about that. I could give it back to our healthcare systems, the ones that renewed and cared for me in a constructive and meaningful way, because I began to hear the data that up to 50% of clinicians experience burnout. So I went to grad school. So let's talk about the underlying causes of burnout. The distinct qualities of a NICU parent leader from a presenter that would be academic, which I am academic, I'm working on my PhD, is that we begin with our story. We're grounded in the value of the experience. So one question I ask you, why did you get into the work that you're doing today? What is your story? We all have a reason for our purpose, for our reason, for impetus to doing our research and our work. So what you'll find in this slide is that there's three characteristics. There's disengagement, moral injury, and a toxic workplace culture, all converging on a system that creates burnout. Notice I said the word system that creates burnout. So often we are told that burnout is an individual experience. It's collective. It is the systems that often create burnout. Now, we have our own responsibilities for it, whether it be self-care and helping reduce burnout. But it is a systems issue. So before we talk about employee engagement, let's go back 23 years. Gallup.com has been tracking employee engagement since 2000. And what they found is that an average of 70% of people are disengaged in their work today. I'm invested in that because that means 70% of the contacts that I have in the healthcare system risk difficulties in caring for their patients. That's 7 out of 10 times. Patients like me are invested in making sure healthcare providers are engaged in their work and changing the systems. So the original term of employee engagement, oh, try that again. You got a sneak peek there. So the original term employee engagement was first developed in 1990 by William Kahn at Boston University. And Gallup has been tracking this since the year 2000. And so let's talk about what employee engagement is. Employee engagement refers to the level of commitment, motivation, and emotional connection the employees have with their work and their organization. It's a measure of how invested employees are, how passionate they are about their work, and how willing they're to go above and beyond to contribute to the success of an organization. Those people who you know are willing to go above and beyond. They're the ones that are going to stay late. Now, I'm not promoting that we all stay late because we have our culture has shifted to now be more inclusive to all people in the work environment. Employee engagement is essential for fostering a positive work environment, increasing productivity, reducing turnover, and achieving organizational success. We create a supportive, inclusive workplace culture, provide opportunities for growth and development, recognize and reward employee contributions, and promote open communication and collaboration between employees and management. Does that sound like your workplace? Engaged employees are aligned with the organization's values and willing to put in discretionary effort for positive outcomes. Disengaged employees fit into two different descriptions. A disengaged employee, they lack motivation, commitment, and emotional connection to their work. Disengaged employees are often indifferent or apathetic. They may exhibit signs of disinterest, low productivity, and minimal effort in their work. They feel disconnected from the organization's goals and values. Disengagement may be caused by various factors, such as lack of recognition, limited opportunities for advancement, poor communication, inadequate feedback, or a toxic work environment. Disengaged employees may have reduced morale and may not proactively contribute to their team or organization beyond their basic job requirements. And if left unaddressed, disengagement can impact the performance and productivity of a team or organization. So what's an actively disengaged employee? This is someone who actively talks negatively about people in their organization. So recently I was talking to a friend of mine, and she had an opportunity to interview with the company and went through the whole process, started onboarding. I mean, was hired for the position, started onboarding, and had an opportunity to do a tour around the organization. And her immediate manager spent 2 of the 3 hours warning, don't talk to that person, don't talk to that person, that person's going to be, and kept saying, this person is a data person. Watch out. This person is critical. This person is, and I sat and talked to my friend and I said, well, do you really want to work there? Knowing about employee engagement is important, because if you are an engaged employee, which I hope you are, knowing the signs and the symptoms not only prevents you from entering into disengagement or active disengagement, but if you're interviewing for a position with a company that shows these signs of a dysfunctional organizational culture, just run. Just run. So here's some data. And I know I'm talking to a bunch of data people, so work with me here. I'm a social scientist. Beyond the breaking point, half of all employees report burnout symptoms. That means half of all employees in every industry. This is a systems problem. This is a situation, organizational issue. This is a issue that collectively that we are dealing with, not just in the United States, but globally. The way our organizations are today, it's not working. We need to find a new way of doing things. What we do know, there are a couple of things that are working. And when you follow the data, when you follow the research, the organizations that are really doing well with low rates of disengagement, that have high rates of employee engagement and participation, is the best places to work. Over 50 to 70%, somewhere in that range, and it changes every year, are highly engaged organizations that are practicing care and concern of their employees. So what is burnout? According to McKinsey and Company, burnout is a syndrome conceptualized from a chronic workplace stress that has not been successfully managed. Feelings of energy depletion or exhaustion, increased mental distance from one's job, feelings of negativism or cynicism related to one's work. Burnout is simply more than just not wanting to go to work. It's a cognitive and a chronic condition that results in fatigue, disengagement, doubt, and feeling trapped and defeated. According to Medscape 2023, the burnout rates among physicians has increased from 42% to 54% in the last 5 years. Now, our immediate thought is, well, COVID, right? Well, when you look at the data, physicians are saying 8% of their stress, of their burnout as a result of COVID-19. Their primary reason is too many bureaucratic tasks, lack of respect from coworkers, too many work hours, insufficient compensation, and lack of autonomy and control on their work. So what is moral injury? It's originally a military term that in recent years, clinicians and researchers like Wendy Dean, MD, or Dr. Z, whose name is Zubian Zamania, have focused on its application among healthcare providers. Moral injury is a psychological construct where a qualified professional is put in a decision-making situation that leads to a violation of deeply moral, spiritual, or ethical beliefs relating to the value of life. Litz defines moral injury as perpetuating, failing to prevent, bearing witness to, or learning about acts that transgress deeply held moral beliefs and expectations. So when Dr. Z presented this in 2019, He said, so many of people in healthcare feel overwhelmed, demoralized, exhausted, cynical, afraid, and alone. We're told we're not resilient enough. We don't work hard enough. Well, it has to be our fault, right? Moral injury, like burnout, has symptoms of demoralization, exhaustion, low productivity, and a feeling of deep cynicism. As humans, we're moralistic, idealistic creatures that love other humans. That's why we get into this work. We get into this work because we love others. And what happens when ideals meet the real world? When we cannot give our patients the care that we need, if we had the tools, the resources, and the autonomy, how would that make anybody feel? Healthcare providers and those caring for patients are some of the most resilient in the world. You made it through med school. You made it through your PhD. For many of you, you are the most resilient in the world. If you're experiencing burnout or you know somebody who is, look at the organizational culture. Look at the systems. So what's toxic workplace behavior? Disengagement, burnout, and moral injuries are the underlying issues that lead to a toxic workplace. Toxic workplace behavior is that interpersonal behavior that leads to employees feeling unvalued, belittled, unsafe, or even unfair or demanding treatment. It's non-inclusive behavior, sabotaging, cutthroat competition, abusive management, and unethical behavior. Statements such as my manager ridicules me. I work with people who belittle my ideas. My manager puts me down in front of others. These are all toxic workplace behaviors. They are often microaggressions and often attributed to a person's lived experience, such as race, color, religion, national origin, ancestry, sex, age, physical or mental disability, veteran status. Anytime a leader, manager, or colleague is dismissive of your ideas in a way that isn't productive, it may lead to burnout conditions. We know that toxic behavior increases burnout. The inability to trust our immediate manager. Increasingly, research is showing that people do not leave organizations. People leave their immediate manager. So what do we do about it? Focus on the organizational culture. The crucial element to burnout is determined by the organizational culture. It's where you work, who you work for, and the people you work with plays a significant role in the symptoms of burnout. Improving organizational culture shifts the responsibility from individual responsibility to a collective whole of behaviors and attitudes in an organization. Healthy organizational cultures do not allow for toxic behavior. Organizational culture refers to the shared values, beliefs, norms, and practices within an organization. Organizational culture shapes the behavior, the attitudes, and the way people interact with each other. Organizational culture is a collective mindset of people and is essential to the way people think, feel, act within an organization. Just as people have personalities and character traits, organizational culture is the personality and character of the organization. An organizational culture evolves or deconstructs within an organization. So you may have a situation where you start with an organization that's very healthy and it's thriving and it's growing and you're engaged in the work and people are so excited for you to be there and you're seeing incredible flourishing. But then one person's hired. We know that one person. And that one person deconstructs an organization. So to improve the organizational culture, this is where the individual aspect comes in. We need to work on our own human skills. These are those interpersonal skills. Some examples of human skills are being relatable or likable, effective communication skills, patience, trustworthiness, empathy, active listening skills, genuine interest in other people, sound judgment, persuasion instead of coercion or manipulation, open-mindedness, honesty, leadership, and motivation. People in organizations thrive when they work with people that they like. And while we all may be a little bit different and we may not like each other, as long as we're respectful, as long as we're willing to approach each other with a sense of honor and commitment, and be willing to talk to each other and say, you know, I don't agree with this data, or I don't agree with this idea, but let's take a look at it. It's not the personalization of, you don't know what you're talking about. It's looking at, this is looking at it as an external feature. So what's servant leadership? Servant leadership is a leadership philosophy where leaders prioritize the needs and the development of their people over their own interests. Servant leadership profoundly transforms the organizational culture to be people-focused over process and financial gain. Interesting, organizations that commit to servant leadership practices increase their return on investment by 3 to 4 times. So do you want to get funded? Servant leadership, creating the organizational culture that is thriving, that is engaged, that has a healthy workplace culture. There's money behind it, there's data behind it to show that money follows high performing organizations. Servant-led organizations rarely have any major layoffs. And when they do, they're usually individual due to performance issues. Organizations that practice servant leadership hire the right people for the right positions, and they're conscientious about the impact of layoffs on the organizational culture. So a little history about servant leadership. Robert K. Greenleaf first coined the term servant leadership in 1970, after a 38-year career at AT&T. He attributes it to a college lecture, his senior year at Carleton College, and he said, one of his professors suggested that if we want to solve the world's biggest problems, focus on the way organizations do business. Focus away from the processes and the performance, or processes and the numbers and the data, and focus on the people. And as we invest in and care for the culture and the people, we will improve the numbers. And so for 38 years, he spent time with AT&T as the first management development manager, taking notes in these notebooks, notebook after notebook after notebook, interviewing people, reading philosophy and psychology, and bringing speakers in to talk with the client, with the people on site. And what he found is that he was the first in the 1930s and 40s to hire people that were Black, Indigenous, people of color, so BIPOC, into management roles. I mean, this was almost 80, 90 years ago, way before it became acceptable. So Greenleaf was concerned about the way things get done in organizations. He recognized the problem of power and privilege. He identified an organizational culture that leads to disengagement, burnout, and moral injury decades before it actually existed. So Greenleaf wrote, I believe that the caring for persons, the more able and the less able serving each other, is what makes a good society. Most caring was once done person to person. Now much of it is mediated through institutions, often large, powerful, impersonal, not always competent, and sometimes corrupt. If a better society is to be built, one more just and more caring, and providing an opportunity for the most amount of people, while supportive of the social order, is to raise the performance of servant of as many institutions as possible by new voluntary regenerative forces initiated from within. That's each of us. So Robert K. There have been 3 stages essentially of servant leadership since it first was coined in 1970. First off, when he coined this term, he was thinking about it from a philosophical perspective. So Lao Tzu, back 2,000 years, 5,000 years ago, and all of the greatest leaders of the world are servant leaders. So we captured all this into a term, into a phrase. So often we're asked to serve other people, serve and serve and serve. We also need to think about that characteristic of leading and making sure that people are getting their needs met, which is also self-actualization and being able to then become leaders themselves. So in 1991, Greenleaf passed away, and the executive director of the Greenleaf Center, Larry Spears, received this box of journals. And he spent years sort of drawing out what are the characteristics of a healthy and engaged leader, of someone who what Robert Greenleaf called a servant leader. And he identified 10 characteristics. So first off, it's putting other people first. Servant leaders prioritize the well-being and success of their team members through a commitment to active listening, empathy, and concern for their well-being. They model excellent listening skills and authentically and actively seek to put ideas from shared decision-making into action. And they track their results. They lead with empathy. Now, there's a difference between empathy and sympathy. Empathy is sitting next to someone and saying, I am here. I'm going to sit beside you during this difficulty. Sympathy is, yeah, that's not me. How often do we have situations where people say, oh, I don't want to go through that? Servant leadership fosters collaboration and inclusivity. Organizations with a high amount of diversity and diversity of thoughts and ideas are typically servant-led organizations. They want to bring all different ideas to the table, including patient leaders like me. They want to cultivate, develop, and bring us into the fold. Servant leaders provide vision and direction. Servant leaders believe in co-creating sustainable, transformative change in society. They set goals. They communicate their clear expectations and help each individual person in an organization achieve success. They build trust and show integrity. Servant leaders build trust in their teams by being honest and transparent, and they model integrity in their decision-making. They lead by example. So that's their reputation. They put their reputation on the line. They foster an organizational culture of trust and accountability. Servant leaders encourage autonomy. If you look back at some of our discussion on burnout, lack of autonomy, and an organizational culture of distrust, these are all things that lead to burnout. So this is the exact opposite. Servant leadership is a solution to the burnout, to the moral injury, to all these issues. Servant leaders model authenticity, and they're brave. When that person comes into an organization that was otherwise healthy, they have the courage and the bravery to put that person through training, interpersonal training and development. They have the ability to say, this is not okay. We're going to either work with this person or we're going to show them to the door. So how do we know if we're acting like a servant leader? So this is what Robert Greenleaf said. He wasn't looking to define servant leadership into a small idea. This was an expansive idea. He wanted to create a vision. Do the people, while being served, become healthier, wiser, freer, more autonomous, and more likely themselves to become servants. At the start of this talk, I told our story about being in the NICU. Our nurses are servant leaders because they created not just me, but all those patients that they cared for who are healthier, wiser, freer, more autonomous. And the few of us that get this opportunity to speak and present as NICU parent leaders are now able to give back to the healthcare systems, the research as research collaborators to improve for other patients. So why does it matter? Organizations that practice servant leadership result in higher levels of engagement, thriving in teams and in their organizations. We all know what happens when you work for a toxic organization. We don't only just experience it at work. It affects our families, our kids, our communities. Same idea. When people work for servant-led organizations and they're thriving and they're engaged and they're seeing this benefit, they're improving their entire communities. They have energy to go and volunteer. They have energy to go mentor. They have energy to go do these things that improve entire communities. So a little about us. That's my son. He's 13. He's healthy. He has a couple things going on. But like any preemie, he's a pretty special kid. And it's because of our NICU experience and the opportunities that I've had now to give back through my own research as I'm working on my dissertation. So this is an entry level. Some of the work that I'm doing as part of my dissertation is looking at the impact of the NICU parent leader and our stories of participating with researchers, with collaborators, with healthcare systems, with academic centers. You can reach me here if you'd like to learn more. For those that are online, I invite you to reach out and ask questions. These are my references. Thank you.
Are there any questions?
thanks for that talk. I was curious when you mentioned there's kind of a list of organizations that have done well with reducing burnout and improving employee engagement. How many of those kind of top performers in that category are hospitals or academic centers? And then amongst those, I guess, what are practice patterns that maybe in your research you found that those institutions are implementing? Because I think that burnout, I'm a physician, so I think burnout with physicians may be very different from burnout in other areas, just for the causes for burnout. So I'm just curious what, if there are any hospitals that are performing well in that area, well, or do perform well.
Sure, I can get more specific information. There is a list on servant leadership organizations particularly, but the things to look at are, what are the listening patterns? Does the executive management listen to the patient-facing physicians? And are they willing to not just listen, but put into action some of that inclusive behavior? Are they willing to make decisions in collaboration with, in partnership with those who are caring for patients? That's probably the number one thing, is empowering and equipping the patient-facing physician to do that work.
How do you, as maybe if you're a junior faculty or if it's not in that kind of C-suite role, how would you enact that change by the people that are making those decisions? I think I could be a servant leader. My role is hierarchy, but how would I get people to buy in, higher up to do those things or make those changes? I think, like I said, it's very top down.
Absolutely.
Myself and my colleagues are trying to do those things. I guess how do you
Well, what is the top down looking for? Good numbers. They're looking for good outcomes. They're looking for good patient participation and engagement. They're looking for the adoption of behaviors that reduce, like I was reading one paper talking about reducing, they were actually focusing in on, and I can provide this paper to you, reducing falls, reducing negative outcomes in the hospital setting. And what they were finding is that when a particular unit is thriving and those numbers are starting to improve, then it's going to get the attention of the C-suite. Sometimes you have to just think of yourself as a unit and improve the numbers in the data that they're looking for, and then it may catch their attention. And then, okay, well, what are you doing? What is this difference that's happening? What is this thriving that's happening? People are coming to work whistling and they're happy and they're engaged. We have a low turnover rate. We have a low burnout rate. We have, all these different things, not just from an employee HR perspective, but also from the way the patient outcomes.
I maybe want to extend on that question a little bit and ask if there's any data for organizations where individuals are very highly skilled, but the senior leadership either has that credibility, has come through the ranks, or is coming from a business MBA background and has never worked in the field of medicine, engineering, some of the other highly technical sciences. Are there differences in the way these organizations perform? Because empirically, I feel that there are, but I'm curious what the data says.
The data shows that people that have experienced patient-facing experiences are better equipped. They have that lived experience to be able to say, this is how things are. This is, they're increasingly aware of the way organizations work. That being said, people that, so I worked for an organization for a while where it was required, it was a servant-led organization, it was required that everyone from the CEO and every executive suite do a minimum of 16 hours a month bedside. Because it's more than just saying, I was a doctor 30 years ago. It's, being at the bedside, of course. It's that staying aware of and being constantly part of that conversation about what the greatest needs of the physicians and the nurses and the things like that are. So how do we stay continually engaged in that? But then also, most importantly, listening and being aware of the circumstances and being approachable, sort of the open door. Having that open door mindset. If someone comes and says, I have a concern, I have a problem, are you willing to say, yes, okay, let's talk, let's sit and talk about it. I believe you. And trusting that person's credibility, professionalism, and experience.
Yes, thank you so much for the talk. I actually recently had a review.
Oh, there.
Hi.
They're right in front of me. I'm looking up there.
Lindsey, Kirkpatrick, hello. A review where, my chair was asking from a burnout standpoint. I'm also a physician in pediatric infectious diseases. So the past 3 years have been very trying. What they can do from a systemic standpoint to help our burnout. Well, I think some part of the burnout is a national moral injury, the sentiment behind the vaccines and misinformation, and not really as much a systems issue from that standpoint. So how, as a servant leader, can you counteract a national moral injury and something we couldn't have changed anything in the sense that, COVID was always going to happen. Maybe our responses could have been different. Maybe our testing could have been earlier, but once it's done, it's done. And, it was in play. Like, how do you counteract that on a leadership level when it's not something that you can control?
Oh, absolutely. Well, what servant leaders like to focus on is how do we heal? So as we come back here, and I'll share this slide with you, get some of the characteristics here. Healing. How do we heal ourselves and our immediate organizations? How do we practice those healing factors? And then as we go through, counselors are great. I love psychologists. I encourage everybody to have a leadership coach and a counselor. Once we get to that place, how do we begin to individually and collectively begin to ask forgiveness? And say, not only am I forgiving myself for having been through this incredibly difficult situation, but how do I forgive just society as a whole for what they did not know? And how do we begin to go through that? It's a very difficult process. I'm not in any way saying it's going to happen immediately, but how do we go through that process collectively? And how do we begin that process of moving forward? And one thing we found is, I was reading a great article yesterday, and they're saying there were 3 stages of servant leadership. The first one was Robert Greenleaf, knows that theoretical idea about organizations. And then the second were these characteristics. What are the characteristics? What are we practicing? This new one is the data. What does the data show around forgiveness studies and around healing? How do we heal collective hurt and collective moral injury? And one thing we like to say is it begins with ourselves. That's why when we did those breathing exercises earlier, that is a servant-led mindset. It begins with ourselves and breathing and healing ourselves, then our immediate families, then our organizations and our community, and then the world at large. It hurts. I've been there. It's a very damaging experience. But how do we love each other? How do we care for each other? Thank you.
Yes.
No, we're good. But being able to come down below someone's level, don't worry, I'm not posing, and being able to see and being able to see them eye to eye, being able to say, I see your hurt. How can I help you heal? It's not going to be done here today at the stage. All I'm doing is introducing. All I'm doing is giving some ideas. We're collectively working towards the need to heal. Any other questions? Thank you.
Very inspiring talk. I hope we can all take something out of that. Our next speaker is the Prince who is a professor of medicine at Vanderbilt University Medical Center and is the PI on the NICE MPRINT CET, Center of Excellence in Therapeutics. He will be speaking on choosing the right journal for your paper.
Thank you. I'm a professor of pediatrics, by the way, but that's okay. Anyway. I'm talking about choosing the right journal for your paper, and right is in quotes because, there's not one right journal for your paper. And the stuff I'm saying today isn't the right way to do things either. This is just one guy's advice. Okay. Victor this morning had a lot of scientific data backing up what he was saying. This is just stuff that I've done, stuff that I tell my trainees to do. And again, take it along with all the other advice you get from all the other great scientists. So I'm actually going to talk about a little bit more than just choosing the right journal. I'll start with submitting an abstract for a meeting, which is an entirely different process. And then choosing a journal. We'll talk a little bit about impact factor and how important or not important that is. I'll touch on authorship, actually writing and submitting the paper, and then your options after rejection, and then going through the revision process. So writing an abstract for a meeting is totally different than writing a paper. Okay, your number one goal in writing this abstract is getting the abstract accepted, not communicating the science. That part will happen at the meeting when you're at your poster or giving your talk. Okay, so writing the abstract is all about getting it accepted. And the key to getting it accepted is knowing your audience. All right, so you're going to write an abstract very differently for a pediatric meeting than you are a pharmacology meeting. Okay. The other thing that you got to remember is the abstract category determines who is reviewing this. So you usually don't give that a lot of thought, but it's one of the more important things you can do. So I'm a pediatric cardiologist. Okay. So if I'm going to submit an abstract on implanted defibrillators in children to the Heart Rhythm Society meeting, I better not choose the sudden death or the ICD category, because that reviewer is going to be looking at, large adult studies that have thousands and thousands of patients. And I've got my abstract with 30 kids in it, and it's not going to get accepted, right? I've got to choose pediatric and congenital EP so that it's a pediatric person reviewing it and they're looking at similar abstracts. Okay, so that's something that you often don't give a lot of thought to, but you really need to. And then writing an abstract for a meeting is a lot like a crossword puzzle. Okay, every character counts. Usually you've got 1950 characters to tell your entire story. So don't waste time saying, this is an important public health problem. Okay, that's nothing you need to spend any characters on. Your intro should be 1 or 2 sentences. So Dr. Thomas had a great intro this morning. She said, breastfeeding is good for babies, right? That's something that everybody accepts. And then she said, I'm going to talk about the role of HMOs in breast milk. And so you can just set the stage with, those 2 statements. And that first statement has to be something that everybody accepts, because if that's controversial or if the reviewer disagrees with it, then you shot yourself in the foot. Don't spend a lot of time on the methods for a meeting abstract, unless it's, new methodology, in which case you've got to explain it. For the most part, people don't care about the method. It's the results that they really care about. So spend a lot of time on the results. And a table or figure usually has a set character count. So usually it's 500 characters per table or figure. So use that wisely. Okay, don't waste a table on a 2x2 table. You can say that in less text than 500 characters. If you're going to have a 5x10 table, that can be a really good use. Similarly, I sometimes see people that have these really elaborate multi-panel figures for an abstract, and that can be a really good use of 500 characters. Personally, I'm doing an abstract at the last minute, so I don't have time to sort of make a really complicated figure. So I don't utilize that as much as I should. And then your conclusion should be definite, but not too bold. Okay, don't say further research is needed, all that stuff that everybody knows. Just say this basically my results support this conclusion. Again, not being too grandiose in your conclusion, because again, that's something that can upset the reviewer. So on to choosing your journal. There's lots of ways to do it. I'm going to go over 2 generic ways to do it. One way is to just write a draft sort of generically, get feedback from your co-authors, and then get consensus on the target journal or a list of journals. So advantages to this method are you can get the best natural fit. Okay, you write the science the way the science is, and then you can sort of figure out what the best journal is. That parent draft can be modified for different journals. If you get rejected initially, you can just revise that for subsequent journals. And your reference list might actually help you decide. So one of my colleagues who is a journal editor says if he gets a paper and it's got 15 references and 10 of them are from the journal Neurology, he thinks, well, this paper belongs in the journal Neurology, not my journal. So if you find yourself citing a particular journal over and over again, that might be telling you this is where my paper belongs. Honestly, I almost never do method 1. I typically do method 2, which is pick a target journal first and then write it for that journal. The advantages of this are it's then tailored to a specific audience. Okay, so again, if I'm writing for a pediatric journal, that's going to be very different than if I'm writing for a heart rhythm journal, for example. Also, the format is specific. So, you write your first draft that's specifically formatted to the journal you're targeting. This is something that Audrey actually told me about. I've never seen this before, but she told me this is here. This is the journal author name estimator. So I've never used this in real life, but I did for this particular example. So you probably can't read the text here. I put in the title of a paper that I wrote with one of our residents, and we just submitted it this week. It's called Association Between Neighborhood Socioeconomic Factors and Length of Stay After Surgery for Congenital Heart Disease, a retrospective cohort study. So this is actually an area that I haven't published a ton in, so I thought, well, maybe this would be useful. So when you click on Find Journals, you'll get a list of journals here. And again, you probably can't read all of them, but there's some pretty reasonable suggestions on here. So Pediatric Critical Care Medicine, Cardiology in the Young, circulation, it's never getting in circulation, but it's a reasonable scope. Annals of Thoracic Surgery, Pediatric Cardiology. So if you have no idea where to start, this is a reasonable resource here. Some of them really, don't belong. So there's, let's see, Journal of Child Neurology. That's probably not a journal that's going to take that paper. And there are other ones too that are very similar. So this is journalguide.com, same basic thing. You can just type your title in and you'll get a list of journals. Again, you can see some of these are the same: Congenital Heart Disease, Journal of American Heart Association. Again, Journal of Neurosurgical Anesthesiology shows up. I don't know what about my title is making people think this is a neurology paper, but for some reason it keeps showing up. So when you're picking a journal, think who you want to read your paper. Okay, does this go to clinicians? Does this go to basic scientists? Does this go to pediatric practitioners, OB practitioners? They all have their different sort of Venn diagrams of journals that they read, and so that can sort of help you pick. Now, impact factor. Impact factor is a pretty easy way to compare journals. It's not the only way though, and so if you're not aware, the impact factor comes out every year, and essentially it takes the number of articles published in the previous 2 years and then divides that by the number of citable items. So that's typically research papers. So not everything that a journal publishes is necessarily a citable item. So it's pretty simple math. It's essentially the number of times an average paper in that journal is cited. Some journals are easier to submit to than others. Okay, some have just a cumbersome process. Some of them try to make it really easy. There's this idea of your paper, your way, where they don't actually have specific formatting rules. you can submit sort of a generic paper. And then if they eventually publish it, then they'll ask you to format it to their specifications, at the time. Some journals are faster than others, okay? A lot of them will advertise, the average time to decision. If they don't, sometimes they'll actually publish on their papers, this is the date that this was submitted, this is the date it was accepted, this is the date it's published, and so you can get an idea. You hate the feeling of submitting a paper and then waiting months to hear anything back, okay? It just really slows you down. So, picking a faster journal is better than others. Some journals will actually have specific priorities. So, you can go to the website and read, about the journal. There's one particular journal that I often publish in that says, we're interested in negative studies because we think that publication bias is a real problem. So, that's a place where you can submit your negative studies. A lot of your journals will also have special focus issues. So, we're having a special focus issue on pharmacology, so you may want to target that. Check publication fees before submitting. So, open access is a thing, and it's not right or wrong. It's just sort of a different philosophy. A lot of journals are very upfront about their publication fees. Some of them not so much. It's kind of buried in the instructions for authors. So, you don't want to be surprised after sort of finally getting through the submission process that, they're ready to publish your journal, publish your paper, and here's the $4,000 fee. So just be on the lookout for that. I always tell people to aim high. Okay. My mentor, Dan Roden, has a quote, I'd always say, well, is it worth submitting, this paper to prestigious journal X? And he'd say, there's only one way to guarantee it doesn't get in there, and that's if you don't submit it there. All right. Aim high enough that you really expect your first submission to be rejected. Desk rejection, that is, the editor just looks at it, says it's not really worth my time. That doesn't take too much time. Usually it's a very quick rejection. If it is reviewed, you actually can get really good feedback. Okay. So, the better the journal, the better the reviewers are. So if you submit it somewhere, it's a high impact journal and you get reviews, you often get really useful feedback. They might take it, right? So this is sort of like real estate. You want to have the cheapest market so the cheapest house in the best neighborhood that you can find, right? You want to have the worst paper in that journal, right? That's really what you're aiming for. And then, if you're expecting that rejection, as soon as you submit it, start preparing your next journal submission. Okay, it saves you time, and again, you're probably going to have to do that if you're aiming high enough. So this is the JCR website where you can, find all the journals and their impact factors. And so at the top, you see a journal that actually primarily publishes review articles. So again, review articles are of course skewed towards getting lots and lots of citations. So journals that publish a lot of review articles will be skewed. But you see, all the prestigious journals: Lancet, New England Journal, JAMA, and so on. So again, I'm a pediatric cardiologist. Here are the top 10, at least in terms of 2021 impact factor cardiology journals. And I don't want you to focus so much on what those journals are, but let's see my pointer here. But anyway, if you look at the number 10, the impact factor is 14. Okay. Now, if you look at the top 10 pediatric journals, by the time you get down to number 10, the impact factor is 5. Okay, so different fields, of course, have different relative citation ratios. So that's something else to be thinking about as you're submitting your journal. Those of you that work in OB-GYN, if you look at your number 10 journal, I think it's a bit cut off there, but the impact factor is 5.3. Okay, so similar to pediatrics, much lower than cardiology. What about pharmacology journals? Well, if you look at the number 10 journal actually has an impact factor of 13.4. That sounds pretty good, but then if you actually look at the journals, you'll find these are journals that mostly publish review articles. Okay, there's very few actual original papers in there. So pharmacology, like pediatrics and OB-GYN, is also sort of skewed low in terms of impact factor. So again, impact factor is not the most important thing. So once you've targeted a journal, you want to read papers in that journal and then write your paper in their style. Okay, it never hurts to cite that journal somewhat heavily in your reference list. Okay, again, the editor will like to see that you're you're citing their journal, that increases their impact factor. And again, it's sort of a hint to that editor that, maybe this paper belongs there. What I typically tell people to do is write, edit, revise the manuscript multiple times with really just one or two other co-authors. Usually it's the first and the senior author, maybe one other person, and get it close to final. And here it's actually very important to spend a lot of time on the abstract. Okay, don't just copy and paste your meeting abstract and put it in your manuscript. This is where the abstract is actually supposed to convey your science, because by the time your paper is actually published, you can probably count on one, maybe two hands, the number of people who are going to read the entire paper, right? There's you, there's your mentor, there's your mom, and then there's, the handful of people that are doing exactly your work that actually care about it all. Everybody else is only going to read the abstract. So this is where you actually do want to convey most of your scientific message in the abstract. So spend a lot of time on it. And then just in terms of getting this submitted, send a near final version to the entire author list and give them a deadline. So this is sort of like the text that I send. So, we plan to submit to Journal X in 2 weeks. Please reply with edits by next Friday. If you need more time, let me know. If I don't hear from you, then that'll be considered approval of this version, okay? That way, everybody has a chance to either, give them their comments, say they need more time, or, or they've approved it. And then you can submit as soon as you're ready. Just a brief word about authorship. Somebody once told me all that really matters are first and senior author papers, okay? First author papers, very important early in your career. When you're applying for your K, the number of first author papers is something that they're going to look at. Senior author papers are much more important later in your career, when you're looking at, how good of a mentor you are, that sort of thing. The corresponding author, it doesn't really matter a whole lot. They're the person that actually does the work of, doing the submission and the revising and everything. But they also may be contacted about this paper years later, okay? So if you are working with your mentor in an area that is not something you're going to spend the rest of your career on, it might be better to let the mentor be the corresponding author rather than you. When you submit a cover letter, and the cover letter is very important in convincing the editor to actually send this out for reviews. So you use this cover letter to briefly state your case. You can say, this paper is novel, or it's the biggest one, or it's the first one to do this. It's of interest to the wide readership of your journal. If it's funded by NIH, go ahead and put that in, because those are all good things. The cover letter is also where you can suggest or exclude reviewers. I don't do this a lot, but actually I probably should, because if you suggest reviewers, then you've made the editor's job easier, okay? And they're much more likely to take it. Also, does the editor have a specific area of interest? Okay, this can be very useful. Once a long time ago, I submitted a paper. It was nothing spectacular, and it wasn't a super high impact journal either. But this was back in the day when you actually used snail mail to submit articles. Okay, so I submitted it, and then I get a letter back pretty quickly. So I was expecting it was going to be desk rejected. It was that fast. But in fact, it was a letter from the editor himself who said, this is an area that I'm interested in, and so I took the liberty of reviewing it myself. And so here's the comments. If you can revise it, then I'll accept it. Okay, so that saved me a lot of time, right? And so again, if you're writing about something that the editor has a special interest in, then that can really help you as well. All right, so now the possible responses once you've submitted it. So there are several types of rejections, okay? There's rejection without reviews, the so-called desk rejection. And that means it's not appropriate for the journal. Either it's not high enough priority, or it's not the right scope. And it's almost never worth trying the same journal again if that's what happens. Sometimes you get just an outright rejection, but with reviews that are favorable. Okay, so it might be rejected for priority, meaning there's nothing really wrong with what you did. It's just not high enough priority for that journal. Or it could be that the reviewers actually had far more negative things to say in the comments to the editor than in the comments to the author. All right, so again, if it's rejected with favorable reviews, but without the option to revise, you're probably better off not resubmitting to that journal. Sometimes it's rejected with an offer to send it to another journal in the family of journals or in that publisher's family. And they always make it sound really good, right? Because they've given you reviews and they say these reviews are will be considered as you revise. And so you feel like you're halfway to getting published in the new journal, but that's not really the case. It goes to a brand new editorial team. They are under no obligation to, pay attention to what the previous editor said. If you do revise it in response to those reviews, they may try to send it to the same reviewers, but those reviewers don't have to take it. So oftentimes you'll get a third or fourth reviewer, which can make things more difficult. And it seems like more often than not now when I get this, the offers for an open access partner. So I've submitted to a journal that doesn't charge publication fees, and they're encouraging me to submit it to a journal that does charge. So, I'm cheap, so I don't like to do that. All right, then there's the rejection, but where they'll consider a revision. Okay, and no matter how non-committal they sound in the letter, this almost always will result in acceptance. Okay, not always, but if they're considering a revision, then they probably won't take it. And so there's other publications on how to respond to reviewers. This is a nice one, 10 simple rules for writing response to reviewers. A key point is the reviewer is always right. Okay, no matter how wrong they are, you've got to consider them right. And so be polite and respectful of all the reviewers. Respond to every point that's raised, and the response doesn't necessarily mean you did what they say. When possible, do what they ask. All right, but polite disagreement, usually with supporting data, is okay if the reviewer is in fact wrong. So responses to so-called problematic requests. Sometimes the reviewer asks you to do more. Sometimes you've already done that and it really wasn't interesting, so you didn't put it in. But if they ask you to do more and you've done it, just say, oh, thank you for the suggestion. We took your advice and we added it. Since it's not important, it's going to go in a supplement. Okay, you can always do that. Sometimes you really can't do it though. Okay, and then again, you can go with, well, we read this would be important, but it's beyond the scope of our current work. Or you can even say, we plan to do that as a future manuscript. And that's something that, it's hard for the reviewer to fight back on if you say you're planning to do it later. What about the problem where different reviewers give contradictory requests? Okay, that's actually an opportunity, because now you can pick the one side that you want and you've got a good reason to rebut the other reviewer. Okay, that's a good strategy for getting that problem. All right, last step, the proofs. Okay, once it's been accepted, you're going to get a request to review the proofs. And this is where the copy editing of your Word document or your PDF is done by scientific publishers. And they're typically, scientific publishers, but they may not know your field specific jargon. So you really have to review everything carefully. Sometimes they'll just change the grammar or usage that aligns with the style of the journal. And again, depending on how it's written, that can change the meaning sometimes. So you really have to be very thorough in reviewing the proofs. And you often have to do this with a very short turnaround time. Usually it's like, we need this back in 48 hours or your paper is going to be delayed. So, be on the lookout for these emails when the proofs are coming and then get to them right away. And this is really your last chance to correct anything. Okay, you can't make substantive changes to the content. But if there's any errors, this is your last shot. Once it's beyond the proof stage, then your only response at that point is a correction, which is something that you don't really want to do. So, and then once the proofs are in, it's time to celebrate because your paper is published. And so that's the last slide I have. I'm happy to take any questions or comments that folks have, or advice from any of the other scientists in the room.
To stop when you you're obsessed with that there's got to be more things to fix, more ways to make it better, and how do you decide, okay, it's time to stop? That's good enough.
Yeah, so when I start, I do this thing my daughter calls vomiting on a page. You just start with just put something down, and then there's a lot of editing that has to go on between that first draft and the last draft. But yeah, at some point, you've got to say, okay, this is reasonable. I'll send it off to my other co-authors for suggestions. That's the point when I stop, fixing every sentence and changing the wording to be just right. By the time I send it out to other folks to look at, I've kind of finished, the minor changes, and I let other people chime in on what needs to be done. Because you can you can spend forever, writing the perfect paragraph, and you don't really need to.
So when you were talking about impact factor, how much does that weigh into the decision on where to submit something to? So a lot of funding agencies and promotion typically are really interested in those high impact journals, like greater than 10. I don't know that I've ever gotten questions greater than 10 because in pharmacology they don't go that high unless you put it in one of those review journals. So how much does that play into your decision?
Yeah, no, it's so it's something that to be honest, yeah, every time I start thinking of the journals, I go to the impact factor rank list and I sort of look at it and say, okay, I know it's not going to get into these journals, but here's one that's just a little bit higher than I think it's going to be. So I do use the impact factor because, again, like you said, it's something that that everybody looks at and wants to be. But then there's some papers that just are in a niche area that is never going to get in a high impact journal, and then again you just want to get it into the best journal you can, and recognize it's going to get into a journal that's got an impact factor of 3 or 2, and that's okay, as long as you continue to do lots of other work and you're putting out other papers as well.
Thanks. I'll also comment that not all journals have impact factors, and it doesn't always make them a bad journal. So new journals don't get impact factors. So I did a lot of submissions to CPT, pharmacometrics, and systems pharmacology. And then when I went up for tenure, had to explain why there was no impact factor. So things like Google Scholar and the H score and the I scores help there too. So. I won't hear you online.
Thank you so much for your presentation. So my question is, what do you think about the journals that are referred to as predatory journals? Because I'm a faculty at the University of Lagos in Nigeria, and I know in the recent years, there's been so much noise around publishing in predatory journals, such that when you are due for promotion and you put up your publications, they have to go through it and exclude those that are published in predatory journals. So I want to hear your thoughts about them, and how do we recognize predatory journals so that we don't fall into that trap?
Yeah, no, great question. So predatory journals are journals that basically are just trying to make money. Okay, they have a very low bar for publishing. It may be peer reviewed, it may not be peer reviewed. If it is peer reviewed, again, the bar is not very high, and so their goal is really just to publish papers and charge people and make money. And again, that's one category. Then there's good journals that are open access, which again is just a different philosophy. It says rather than charging the end user, to pay for our journal, we're going to charge the authors that are publishing. And there are very well respected open access journals, and those are fine. How do you know if a journal is predatory or not? If you've never heard of the journal before, then that's probably a red flag. If it's a journal that charges, if it's a journal that doesn't charge any fees, it's not going to be a predatory journal. If it charges fees, that doesn't necessarily mean it is predatory. And then again, you just have to rely on. If you've never heard of it, you've got to rely on other scientists, and ask them, is this a respected journal? Do you recognize some of the names that are, published in that journal? Is that worth publishing? But yeah, if there's any suspicion that the journal is predatory, don't bother. There's always another place you can go. Did that answer your question?
Let you read it.
There's a question from Rupsa online. Can you discuss balancing time to acceptance with aiming high?
Discuss balancing time to acceptance with aiming high. Yes. So it's pretty rare that there's actually a race to publish. Okay. If you're doing something and it's a super competitive area and there's other investigators working in a similar area and you really have to get it out there, then yes, you might not aim quite as high just to get it published. But that's pretty uncommon, honestly. The other time that you don't want to spend too much time is if particularly if there's a trainee that's then, moving on to another job that may not be academic. Once they leave, they're not going to spend a lot of time. And so, at that point, you want to just get the paper out. The rest of the time, there's usually not that much of a delay in aiming high. Like I said, if they reject it, without reviews, it's very quick. Even if it gets reviewed, it's usually not that long anymore that you're delayed by aiming high. So I will typically, err on the side of, aiming high, recognizing I might be delayed a little bit, again, except for those two rare exceptions.
All right. Are there any other questions, comments? If not, join me in thanking Prince for his talk on choosing the right journal for your paper. All right. So we have a 15-minute break. So stretch, relax, network, ask questions, and then we'll come back for a panel discussion focused on career development. Thanks.
I'm live. Well, everybody, welcome to the final session for our trainee day of the MPRINT Hub meeting. So I'll give a quick introduction for those that don't know me. I'm Aaron Pollack. I'm chief of the obstetric and pediatric pharmacology and therapeutics branch at the Eunice Kennedy Shriver National Institute of Child Health and Human Development. I do practice saying that. So this is what this type of event is always one of my favorite to go to, because really I think the trainees here, you are literally the future leaders of our field in multiple fields. So it's really exciting to meet you all at this point in your careers. I really love the science today, the breadth that is engaged by all of you here, and the fact that, the vision of the MPRINT Hub being able to help and enable trainees, both in terms of support, research, and then events like this. I'm really glad to see this come to fruition. So really, we're going to flip that a little bit today. The panel we're ending up with now is really on career development. So what we have are some folks that'll be available to talk about their careers. And then we want to hear from you to sort of help address questions you have. And, we can be pretty open about career questions you have as you think more about what you want to do in your future careers. So I'll just take the liberty. I'll also give a little bit of my background, and there's more expertise here, so we may tap into folks as well as questions pop up. But I of course came up in academia. Actually did my PhD in enzymology. Went back and worked in a lot of different therapeutic areas as a postdoctoral fellow. Went into the pharmaceutical industry and biotech for about 5 years. Then transitioned to the NIH, originally at NIDDK, being a program officer. Really, I really enjoyed the pharmaceutical industry and drug discovery. Wanted to go someplace that had a little bit more long-term strategic vision, right? Being able to think of and see things like the MPRINT Hub come into fruition. And that really drew me to government and NIH work. And then transitioning to NICHD to be branch chief and sort of take on more of a leadership role with program directors. So that's sort of what has led me here today. So I'm going to start to my left, and hopefully folks can kind of tell their journey of how they got here today. Leslie?
Absolutely. Hopefully everyone can hear me okay. I see some confused faces, so we're not sitting in order. That's listed here. So I'm Leslie Samity Bates. I'm a program officer at NICHD, same branch as Aaron. I also was pretty much brought up and raised in academia and always thought I would stay in academia. I am a pharmacist by training, a PharmD PhD. I also did a clinical pharmacology fellowship at UCSF. So California is pretty familiar to me. I actually transitioned to government after realizing I wanted to have more of an influence in the training process. I really liked my training, and I just wanted, what I realized is not everyone was afforded the same opportunities that I was, and I wanted to sort of even the playing field. And I think joining the branch and Aaron allows me a lot of latitude in terms of really pushing the training envelope. So yeah, I think I see some familiar faces. I lead the T32 ClinPharm Fellowship at NICHD.
So good afternoon. My name is Sharon Nachman. I'm the division chief for pediatric ID at Stony Brook, and I'm the PI and chair of the Impact Network, which is the maternal child HIV and TB Okay, I got the message. I'm sorry. The Maternal Child HIV TB Clinical Trial Network, and obviously very excited to be here among such distinguished colleagues. I wanted to say, you know, it's always interesting. I'm full-time academics. I've been at the same institution for over 30 years. I have the gray hair to prove it. But I think it's more important to say a little bit about my story is that I'm a first-generation American. I'm the first in the family to go to college, obviously the first in the family to go to med school because, you know, we didn't have any of those. And, you know, English is a second language in the house I grew up with. So that's a little bit different. Most people think of academics, so we're so ivory towered and you had your parents and your grandparents and they were doctors for generations. And the answer is no, it happens sometimes differently. And you know, certainly when I was in med school, there was no one at home that I could discuss anything about med school with because no one went to college, had any clue what I was talking about. So certainly academics was a reach. And for many years, my mother would say to me, so when are you going to see patients? When are you opening an office? I'm not. So the way I got into what I'm doing is just by being, I guess, obnoxious and stubborn. I think that's a trait some of my colleagues in the audience are going, oh yeah, that's her. And I always was bothered by the why question. Why do we do stuff? Just because someone told me to do it doesn't mean it's the right thing to do. And over the years, that got me into a fair amount of trouble because I would, you know, sit there and say, just because you said that doesn't make it true. I need to understand why that happened. And you ask it enough times and people give you the work to say, okay, you go ahead and do that. That's fine. And I came into clinical trials because HIV happened at the same time that I graduated from my residency. So that juxtaposition of a new disease and everybody was making statements about something they had no idea about led to a group of us very, very, very young investigators in the back of the room heckling the speakers. And that translated into they gave us work to do because they didn't want to hear from us. And we took that and ran. And now through the years, I went through the growing up of the network to become the chair of the network. And now in my 10th year of running the network, which gives me even more gray hair. But just to tell you that just because someone tells you that's how it's going to be, doesn't make it true.
Done?
Done.
Good afternoon. I'm Gil Burkhart. I'm Associate Director for Pediatrics in the Office of Clinical Pharmacology at the FDA. I did my PharmD in residency at the University of Kentucky and at Norton Children's Hospital in Louisville. And then so my first faculty position was at Buffalo. Back then it was the State University of New York at Buffalo. And it was Buffalo Children's Hospital. And moved from there to Le Bonheur Children's Hospital in Memphis, Tennessee, University of Tennessee for a while. Then so I learned a lot in Tennessee, but so I was able to apply that when I moved to the University of Pittsburgh. So I was in Pittsburgh for 20 years. I was very fortunate to move there at the same time that a guy named Tom Starzl, who was the father of liver transplantation, moved there. And so I did transplant studies at the University of Pittsburgh. I was NIH funded for almost 20 years at Pitt. And then I decided I would try out academic administration. And so I moved to the University of Southern California in Los Angeles. I was department chair there. And fortunately, had a chance to do a sabbatical at the FDA with Larry Lesko in the Office of Clinical Pharmacology. That was in 2006. And then in the fall of 2007, under the FDA Amendments Act, basically the FDA, I could see the legislation that was coming in terms of pediatrics and with the formation of the Pediatric Review Committee at the FDA. So they offered me a position. And so I moved to the FDA and started there in February of 2008 and as Associate Director for Pediatrics. And so I was in academia for 33 years, and I've been at the FDA now for 15 years.
My turn, huh? Good afternoon, everyone. My name is Sasha Ren. As you can tell my name, so I originally from China. So now I'm the program officer at the obstetric and pediatric pharmacology and therapeutics branch at NICT along with my colleagues here. So I came to United States to pursue my PhD, which I went at University of Alabama, Birmingham, got my PhD in cellular and molecular biology. After that, I did my postdoc at the National Eye Institute at NIH. And then, as you can tell, actually, I was a physician before I came to the States. I was actually practicing physician as an ophthalmologist. So all my research before that is all related to the eyes. So it's kind of interesting. I'm moving to different space. But anyway, so I finished my postdoctoral training and I went to a informatics company briefly a little bit more than a year. And then I found that I'm not enjoying, so I think that's not right place for me. So I had a great opportunity, came back to NIH. So starting from there, then I began to work as an administrator, so as a program officer since then. And then I joined NICT in 2008. So as a program officer since then, so all the years, I think that been working at NIH extramural for more than 20 years. Yeah, so as you can tell, I have very broad program background, so moving from different fields. Yeah. So I'll stop there.
Great, thank you all. So you can tell we've got a lot of expertise here, a lot of different paths that people have taken. So want to open it up to questions now. So anybody that has a question, raise your hand. Also keeping an eye on the chat for those that are in virtual attendance.
The questions always start on the opposite side of the room.
So my question is for Leslie, who's presumably the youngest on the panel, but and you also happen to be working in a role where there's the scope of COI concerns is relatively high. So how do you advocate for yourself from a professional development standpoint to get experiences that your colleagues outside of NIH may get that will allow you to grow in the confines of a very high COI organization?
I'm still navigating that. I think parents that it best, I just put a little practice and persistent that sort of thing because I want to do it. But I mean, we also have to have really great leadership. Like I have a parent who's very open, so pushing
me in my career. Oh, excuse me. And I think it's just asking those questions, right? The worst thing, I've always been told the worst thing you can hear is no. And if you don't ask, you'll never know. So I always am trying to reach out to figure out, what things I can possibly do that won't be in conflict with NIH, or if there's ways to kind of get around that. So, I don't think I have one answer for that. I feel like I'm still navigating that. And the way I do that is by asking the questions. And is it possible? It's sort of hard in the government, I will be honest, because you can run into some walls. But just know that there are opportunities to sort of push your career if you really want to and are really passionate about it. You just have to be persistent.
Yeah, maybe just one thing to add to that, there are certainly a lot of complexities to working with the government and different sets of rules between the NIH and the FDA. Another nice beneficial thing about being in the government is, although it could be incredibly bureaucratic, one perhaps upside to that is once you're in there and you have experience, you have a set of skills that really nobody from the outside has. And there are certain positions in the government that you can move into that would be difficult for anybody that doesn't have that experience to move into. So that's an additional layer or aspect of career development that we can take in the government.
Can I just respond to something you said, Eric?
Absolutely, guilty.
About the government being bureaucratic. And that's true. But academia, if you're in academia, it's just as bureaucratic. Now, the emphasis is on different things, but it's just as bureaucratic if you're going to be a long-term faculty member. Let me tell you.
So I was going to say, I think that industry is perhaps even more bureaucratic with its multiple layers and table of organization and who signs to who. So I think anything that we do that we're going to be successful in, we have to recognize there are rules to it. You have to learn the rules and you have to abide by the rules and understand how do they apply to you. And then I want to say politely, do the best way that you can so that you're successful.
It helps to be a little more mature in government because you pick your fights.
Yeah.
You know, early in my career, I probably, I wouldn't have been patient enough to take it, but now you kind of sit back and say, oh, okay, I can, let that go.
I do see one question from online is what are ways to be involved with the NIH, NICHD without physically being in the DC area? That's a good question. I'll try to answer that a little bit. And I think Gil could also have an FDA perspective on that. So, there are different opportunities to be involved. Certainly we sponsor a lot of workshops, a lot of activities. There are occasionally opportunities for volunteer folks that want to do work within the government, not just the NIH. There are also a variety of different government fellowships. The American Academy Association of Sciences has a fellowship opportunity. There are a few other ones that allow people to come in for a year or two. And in the pandemic and post-pandemic era, we are looking at more virtual options. We've actually hired people, a fair number of people in the NIH that are full remote work. So there are some opportunities for that. And Gil, I know the FDA has a fellowship department in your office. Can you comment on that?
So was the question specifically about how much you can do virtually?
Yeah, without physically being in the DC area.
Without being in the DC area. Well, actually, of course, we have a lot of experience with that now, with the pandemic. And most of the work of the FDA has actually progressed very well over throughout the pandemic. And we had actually prepared for that in a way because we ran out of space at the FDA. And so basically the staff was working from home half-time before the pandemic. So the work actually went okay, but I told people the things that didn't work as well were the teaching program with the students and fellows. And so yes, we have students and fellows who have been doing, virtual rotations with us and working on projects and go to our meetings and attend the FDA meetings. They get a computer and a badge, and so you can do all that. And I just don't feel as good about having the ability to relate very frequently with the fellows, kind of sit down and talk to them and have a hallway conversation, those types of things. So that hasn't been as satisfying. But in terms of doing it, yes. And even if people want to talk about, positions with the FDA, they have a category of people that just work 100% remotely.
Actually, Sharon, as an academic representative, I'm actually curious, how has the virtual work affected the academic world and the functioning of networks?
So I think it's been very difficult. I think that while we have transitioned to Zoom, and I want to say I only know the top half of many of the people in the room, I actually think we've lost the ability to really do a good job on the discussions at the water cooler, for lack of a better word. The kind of stuff that people in the hallway say, hey, I have a question for you. What do you think of it? I think we've lost that. We've moved back in many academic institutions to having people come into the office. But what's interesting is more departments gained more faculty. There was no place to put them. So all of a sudden, people in positions of power were excited that people weren't coming into the office because we didn't need to give them an office anymore. So I think when I think about our young faculty that came in during COVID time, I actually think they've lost something. I think there is something very special about being in that environment where you have 10 people right next to you that you could chat with and talk to that weren't just a Zoom call. So I'm hoping we are moving back to more in person because I think academics and a lot of the questions that come up are not straightforward.
We're requiring for our students that come on rotation with me. For my group, we have about 18 pharmacy students per year. And we're requiring this year that they come during the first week at least and be on site. And so that allows us to do some technical things in terms of getting them their badge and computer, but it also allows us to have some personal discussions with them, discuss their projects and other things. And so we're requiring kind of 1 week on site at the start of usually a 6-week rotation.
All right.
Question. I was curious. So there's a consensus that we all live in a very highly regulated environment, no matter what environment we are in. So, as you started out in your careers, were there particular insights, particular strategies that helped facilitate navigating successfully that besides the persistence and grit, I suppose, but was there other strategies or other perspectives that helped facilitate your success in those systems?
So I can talk from the academic side of things. And I think part of it is, recognizing that there are stuff that's out of your control. Part of it is getting some good mentors that you can ask some of the questions to and say, hey, I'm having this problem. Here's my, what do you think? And I think part of it is also coming up with novel, creative solutions on your own that may or may not fit. And, sometimes it's like throwing the spaghetti on the wall, something will stick. So there's no one right answer. You're going to see lots of, walls in front of you, and your job is to either get help climbing over them, destroying the wall, growing right through it, or going around it. And no one solution fits everybody at the same time. So I want to say, recognize that they're there and don't pull your hair out. Sit there and think about, all right, how am I going to fix this?
So I would second that and say that colleagues, mentors and colleagues, those are the people I look for academic institutions. Those were the people that when I think back about those experiences, it was always the colleagues, particularly, a mentor certainly early on, but later it was colleagues that I could relate to and work through problems with, and that was really did facilitate my career at each of those institutions.
So, it's funny, during the early HIV epidemic, as you can imagine, we had no drugs, we couldn't diagnose kids, and they died. And it was a pretty, as bad as you thought COVID was, the early HIV epidemic was not any better. And we had a group of us young faculty across the country that talked once a week. And it was our, complaint session once a week. We did the conference calls. We had to dial each number, 17 digits long. You screwed one up, you had to dial it again. There was no Zoom. And it was very, very helpful because it, I think, prevented some PTSD amongst all of us who basically would make rounds, and you'd see a child and go, all right, they have HIV, they're going to die. That's not good. And then I have to give the mother that bad news, you have HIV too. So I think somehow pulling together a group of colleagues that are not necessarily your institution, but are similar thinking about stuff that you're doing across the country, and setting up that you actually talk to them, not just when you have a problem, but when you don't have a problem.
I have a slightly different strategy experience to share. So like personally, so when I was a graduate student, so in the lab, I think everybody has the same experience. So hearing the mentor applying for NIH grant, so when your mentor got a grant, so I think every lab does that. They celebrate the opening champagne, and so at that time, So not too long. So at that time, I saw my mentor so excited. So I didn't know how important it was to get an NIH grant, but I was fascinated. What what kind of NIH was that time? I wasn't sort of didn't truly understand. So until I had a chance to went to a seminar, actually was the speaker was actually from a National Eye Institute. So, and then I listened, and then after that I was discussing with him, and then I said, I'm about to graduate. I'm looking for a postdoc. So he offered me immediately. He said, send me your CV. So then I said, I was one because it's fascinating about NIH. So finally got a chance. So, going to NIH, get my postdoc. So from then, and then I still not quite understand because NIH postdoc sort of intramural equivalent to academic, still didn't quite understand what was one application was about. So with that kind of sort of curiosity, until I had a chance to work for extramural, so truly understand what the grant process and how NIH, grant application it was. And then I thought that, I was actually lucky. I'm working at the other side, but had the passion, try to help people to apply for grant because I knew how hard it is to get an NIH grant. So, but that was sort of my personal experience story. So I was sort of fascinating, sort of interesting, sort of like behind my back and my mind, I was always wanted to explore what NIH is. So I think since then, sort of you set up your goal. So that may be your place you wanted to be for your career.
I have a question. Earlier, Audrey shared of a mistake that she made related to the social media presentation we had earlier. And it was really interesting. I was wondering if you would be willing to share a mistake that you made that maybe shaped your career or impacted your career.
Yep. Well, I think it was for all of us.
Who's first? Jen? Sorry.
Can you repeat the question?
Can you repeat that?
Yeah. So Audrey shared a mistake that she made that kind of impacted how she interacts with reporters and media. And I was just wondering if there was a mistake that you have made in your career that got you to where you are, and how it maybe shaped what you do that maybe was not a mistake eventually, or something that you learned from. I know it's kind of a hard question. Sorry.
I can't really go for it. And I guess I'm struggling with this question because I appreciate the quote unquote mistakes that I've made. They kind of helped me and guided me to where I'm at. So that's why I'm still thinking about it. So maybe come back to me and I can.
Yeah, I'll volunteer to jump in here. And maybe, modeling authenticity as a leader is part of this. Yeah, well, I was moving around. So I worked at biopharmaceuticals for a while, and I got affected by the layoffs when Pfizer acquired them. But I jumped around a few biotechs, so I'm not going to name names. But there was one, I took one job, I was looking for jobs at one point. I took a position, and it was the first time in my life where I took a job, and on the first 2 or 3 days, I was like, oh my God, and wow, yep, this was a bad mistake. This was a mistake. That one, realizing that quickly is important, and owning it, not, ostriching and burying your head in the sand. I won't detail all the flags that I saw afterwards, but I was sold something that was not what was actually there. It was basically what it is. But at that point, it's like, well, you're here, what do you do next, right? So first of all, getting past I made a mistake. Really helped me focus on, well, what do I want to do next, right? And then engaging, finding new people to help guide me and to ask questions to, and really just trying to use that as an empowering way to make more informed decisions in the future, but also, just to really explore different options. So when you do get into those kind of situations, first of all, not easier said than done, but okay, it's done, the decision was made, you've got to figure out going forward. So that was probably one of the biggest mistakes, if not one, the biggest I made in my career. But 15 years later, in a very good place.
So I'm not sure it's a mistake, but I would say be careful what you do for money. So when I was at the University of Pittsburgh, and I was in pediatrics, I was in the Pittsburgh Children's Hospital. And in the ICU, we were taking care of liver transplant patients. And I was, at that time, I was ready to get some NIH funding. But pediatric studies were not fundable. And we just didn't have sufficient numbers. And so I really had to work in the adult realm. And so that's where my grants were. That's where I had to put a lot of my effort. I eventually actually became the research director for the Division of Cardiothoracic Surgery, but it was all adult stuff. And so it was actually part of my move to the FDA was to get back into pediatrics. And so the organ transplant stuff, I really enjoy it. I think we made a nice impact on patient care, but it took me away from pediatrics.
So after I finished my clinical ID fellowship, I applied for a physician scientist training grant, and I guess luckily or unluckily, I'm not sure, right, was awarded one. It was when they were very first starting out, and I got a postdoc position at Rockefeller. And Rockefeller is a research institute, and I was working in a lab there. And coming from a clinical PEDS ID to a basic science lab was a very big jump for me. And similar to what you said, after being there for a while, I was like, this is really not for me. But I stuck out the 3 years. I completed my physician scientist training grant. I wrote the papers, and what I learned from it is what I like, what I don't like, but more importantly, it was a way of thinking. In clinical medicine, you get a CBC and the child's anemic, you say, okay, I'm going to transfuse you. In the lab, you get a lab result, and the first thing you say is, okay, what did I do wrong? How am I going to repeat the experiment with 4 different variables to actually get back to, did I think I started it and set it up the right way? And that type of thinking actually influenced me quite a bit in academics, because I wasn't going to respond immediately to a result or a lab or a clinical trial or anything like that, but rather sit there and say, do I really believe this? Was it done correctly? Did I ask the right question? And if I didn't, how am I going to ask it better? So I think that while the lab time was not my favorite use of my daytime hours, it actually taught me quite a bit more about how to really think about a problem that you wouldn't have gotten just coming from a clinical ID fellowship.
And also, I just wanted to echo, it's not a mistake. I think it's the passion, no matter where you're gonna work. So for me, first, government versus industry, I mentioned earlier, I had, briefly a little bit more than 1 year experience after postdoc working at a industry. So I didn't enjoy that type of environment. So I could stay because it makes more money than, government. But, it just really depends on your personal preferences and also other factors, your family and all other. So, and the location, I really like the location where I am, I enjoy, close to DC area. And that's also a major factor as well, make decisions. So where you're going to end up, settle down. So, and I think there's so many things. So, of course, you have to make a combined decision. And so, yeah, there are some frustrations working for the government for many years, but I'm sure everybody does, no matter where you are, it's the same thing. But once you decided to stay, you sort of learn that, adapt to the environment. And now, so yeah, I really enjoy what I'm doing right now. Happy to see, I can help my grant team, to get funded. So that is actually my enjoyment. I sort of achieve something I can at least help them. So yeah. So overall, it depends on, you have to have a passion, no matter what you do.
My only comment would be not to dwell on the mistake, right? One thing I've learned from I mean, the panel and, you know, a lot of my mentors is that it doesn't completely derail you, right? That's something that happened and you can move on to something else, right? You're not stuck in that mistake. You may be stuck in it for a little bit, right? But there's always an option to move on, to move to something different. So I think for me, I like a lot of my mistakes. I can look back and say I tried it, it failed, and I moved on to something better. Maybe I stayed in a position a little bit too long. Maybe I stayed in something perhaps for the money, you know, but I realized like, okay, this is not for me. And I was able to jump out of it. So I think resiliency is kind of key here. You know, don't dwell on your mistakes, just try to sort of learn from them and move past them.
The question for the whole panel, and it's a response to something Sasha shared, which is you got your fellowship because of somebody you met or somebody you knew. And so for our advice for our young trainees here and our young faculty, how much of their next job, getting their next job, is going to be who they know, not what they know, and how important is networking going to be for them to get that next job?
I'll start first. So I think now because we're in a different era, so you know, information is so much in advance, and technology so much in advance than we used to be. So there's so many opportunities for the, you know, the fellows and students. So I think that they should take advantage of the current information and technology. Try to, you know, well, first of all, you need to set up your internal goal. What do you want to be, you know, for your career? Then try to expose yourself to be, you know, talk to as many expert as possible to, and for me it's just I think it's just a little bit I think a unique case because I had a chance to met my mentor, you know, by seminar. I think that sort of you could use that analogy. When go to as many meetings as you can to expose yourself and to express your interest, or now you can, you know, there, as I said, a lot of other ways to communicate. So that will make you, you know, a lot of opportunity for you to find your dream job and or for next step for your career. So I think networking is another key skill as well. And again, to try to expose yourself as much as possible.
For FDA, when we have positions available, they're posted through USA Jobs. Well, if you apply through USA Jobs, it's you and 10,000 of your closest friends who are applying for those jobs. And so it's really difficult for you to have a good shot at that position unless we somehow get to know you. And so how do we get to know you? Well, you know, we have T32 fellows who actually do rotations at the FDA. We've had several that have worked with my group that have actually been taken staff positions at FDA. We have an ORISE fellowship at FDA, you know, which is really very much directed toward people who ultimately are thinking about having a career in the FDA. And it's one way we get to know people. I run a summer internship for students, both PharmD, MD, and PhD students who do 8 to 12 weeks during our summer internship. And you know, and Jim, it's earlier in their student program that they come, but it gives them a chance to see what we do, and it gives us a chance to get to know them too, and kind of sets them up, I think, later for having a position in the FDA. So you hate to say it's not like somebody it's you know, it's not who you know, but it is to a certain extent, at least, getting some experience with people in the agency so that you can get weeded out of that pile of CVs.
So I want to take sort of the opposite piece, what you shouldn't do. So invariably, you know, in academics, I get emails all the time from people who read my research and want to work in my lab. Well, my lab is my telephone and my computer. There is no lab. It's just me sitting at the desk, thinking, typing, calling people and writing. That's the whole lab. So if you're going to email to someone that you would like to work with, be smart. Look who they are. What do they do? What are the things that they're thinking about? And then develop a question for them that you want to work with them and why. You're interested in this, and that's what they do, and you read their paper, and can you collaborate with them? Can you work with them? Can you meet them? Can you talk to them? And if you do want to meet them, don't be late. The worst thing that happens if my time is valuable, I expect everyone in this room's time is valuable. And if you have an appointment to show up at 10 o'clock, be there at 10 to 10, not 10:15. You've just pissed me off, I'm done. So I want to say that's sort of the things not to do. If you want to work with someone, research who they are. What have they done? Where have they done it? What are the things they're thinking about? And then send the appropriate worded letter to them. And the other thing is that I'm very excited we're not Zooming, because I really like to do the talk in the hallways at meetings. That's how stuff happens. And also, it's who you know, and you've met someone and you say, hey, I have a junior faculty who wants to do this. Can we get them something to do in your HIV network? And the answer is, sure, let them call me. And if that junior faculty then follows up and calls me, oh, they're ahead of the game. We'll find them something to do.
Yeah, I would agree with what everyone said on the panel. I think, I guess just to add to it, I'm really big on sponsorship and finding a good sponsor, not just a good mentor. You know, all your colleagues are brilliant, right? You're pretty much, I won't say on the same playing field, but yes, pretty much everyone is brilliant. Everyone is smart. Everybody's bringing some great ideas, but really having your name out there and being recognized, right? When you submit an application or submitting for a talk is, it works wonders. So the one advice I have to use, yes, have a great mentor, but also have a great sponsor, someone who's going to put you in the right room, someone who's going to offer you opportunities. For me, I'll give you an example. My PI, my mentor, my sponsor, I always told him, I was like, I need a platform talk. Like, I need someone to know my name. And he was like, well, you know what? I have to speak at the FDA, you can have it. And I was like, you know, that was eye-opening. And I got so many emails afterwards from, you know, people at the FDA, people at NIH saying, hey, I heard your talk. It was really great. And I was like, wow, as a fellow, you know, that was really big for me. So I don't want to say it's who you know, but I think it's all about how do you get people to know who you are. So just kind of keep that in mind.
Yeah, and another aspect to that, I think, is something, you know, a term informational interview. You know, it's a little bit not interviewing for a position or asking for a position, but just asking somebody to talk to you about what the career is like, what the space is like. And that's sort of a softer way to get an idea of what's going on in that sphere, also to make connections that, you know, so if that goes well when you are looking for jobs, you know, that's a good way to build a network. And also, I think, you know, our CVs tend to get very big. I'm not necessarily a big fan of a resume, but I think cover letter, like Prince was talking about the abstract, that's your selling piece. You know, being able to write a succinct cover letter for jobs is another way to make sure that people see the highlights. You want it very well crafted, that sort of gets around the who you know. It's just being able to put that document together. Online question from Shirley.
Thank you. So all of you have been in, you know, maternal pediatric research for many years. And so for some of the trainees in the room who might be thinking about academic careers, what do you see as the next challenges in maternal pediatric clinical pharmacology? You know, what's the next frontier? What are the questions that our trainees should be asking?
Everything. Yeah. You know, I want to say what we know about maternal pediatric pharmacokinetics, pharmacogenomics is about, you know, 1%. And it's only a little scratch on that 1%. And, you know, I'm fortunate that some of the people in the room here, I write clinical trials with them. And, you know, we talk about all the time what we don't know. And that far outweighs what we do know. I want to say the field is completely wide open. You know, the joke is we have so many medications that are approved for children with no PK data. And we blindly just give them and say, oh, that's right, that's the dose. We have no data. And when you look at maternal pharmacokinetics. We guess even more in the HIV TB space. We've had to do every single antiretroviral, every single TB drug, multi-drug resistant TB drug to get an idea of, can we dose it? What's the dose? Second trimester, third trimester. So I want to say politely, the field is incredibly wide open because we have more questions now than we ever did before. And, we're trying to write clinical trials for how to dose neonates. And that's a whole new ball game because our models for neonates, are confounded by how old is the kid? 4 hours, 2 days, a week. When we had to develop the dosing, for example, for raltegravir, which is an antiretroviral approved in adults, finally approved in children, 4 weeks. And then that first 4 weeks, we had to, do dosing tables that were dependent on the hour of how old the kid is. And, we talk about it, but there's no parent out there that's going to be doing that. So we have to think of other things to do. So I want to say the field is wide open. We know very little. We have great modelers out there doing incredible, fascinating work, but we need to do more.
Yeah, and when I think about sort of gaps and opportunities in the field, I saw there's a presentation I saw when I first joined the NIH at one of our career forums, but it was Alan Siltiel, works in type 2 diabetes and obesity. But he gave a great presentation that was a little bit, setting up a dichotomy of, people are either tool users or tool builders. Like, yeah, people can do both, but they're predominantly fit to one or the other. And that was a light bulb moment for me because I feel like I'm very much a tool builder type person. So as you think about your career, that might be one thing to also just, reflect on yourself, because there's a lot of opportunities for new tools and new ways of doing things to generate data as well as conducting those studies. So that's something else to think about.
So clinical pharmacology is very quickly becoming the science of integration of knowledge to affect drug development. And so, so I'm not a modeler, that was, but I understand. I feel like I understand modeling. It's changed what we do in pediatrics in terms of drug development. It's still changing it, where we have multiple conferences this year, for example, on drug absorption and understanding drug absorption, being able to model drug absorption in even neonates. So, so the knowledge is evolving. It comes from human studies, but it also comes from animal studies. That's most of the maternal and lactation studies or animal studies. Comes from that. Comes from in vitro data. Comes from a systems pharmacology approach, and so just don't ignore that. You don't have to be a modeler. But just don't ignore that part of the science because it's becoming so important. You have to be able to be a user, or isn't that what you said? A user instead of a
user builder.
Yeah. Okay. So you have to be able to use that part of what we're doing. It's become a huge part of drug development.
So a lot of you have had relatively non-linear career trajectories. And I wonder if you can talk about kind of the difference between having a career goal and being like, I want to do this type of work in this type of institution versus really being open to new opportunities and trying things that you maybe hadn't considered when you were first sort of plotting out your career.
I'd say keep an eye open for opportunities, I've had some colleagues, they're they're being celebrated at the university because they've been there for 40 years or whatever. And I thought sometimes I think, oh, that might have been very nice. But excuse me. But I always look for opportunities, and that's why I ended up moving around a bit. And I think that's a positive.
I want to do the con on that side. I think it's within you. Even if you're staying like I did at one academic institution your entire career, what I did 10 years ago, what I did 20 years ago is quite different than what I'm doing today. And there are opportunities that you have to make for yourself. You have to, I tell I tell our medical students, I am not your mother. It's not my job to make you happy. It's your job. So that applies for our academic careers as well. You have different opportunities and you have to create those opportunities. And they could be within your institution, or they could be, 5,000, miles away at a different institution. And it's up to you to decide what fits for you at the particular age that you're in, or the career or time that you're in. I have 3 kids, and what worked for me now would not have worked when they were quite young. And I did different things when they were in grade school and I had to go to, class plays and carpool and stuff like that. And they're different now that they're out of the house. So what you're doing is up to you. And those career changes may be at your own institution, those opportunities, or they may be at a neighboring institution. But again, it's your job to figure out what fits for you at that time in your career.
Yeah, and I would say, like, for me, on one view, my career path is non-linear. I think many people's are not, but it also, it's careers in life is a multidimensional space, right? So if one were to look at the vector of being passionate to have impact on health, then it actually has been a pretty linear career. It's just taken a different path at different times, and a lot of that depended on where I was in life, sort of what I wanted to do next, what I've learned, and opportunities and challenges. I've often been motivated by, all right, I've been doing this for a little while. I want to do something else now, just to take on a different challenge. So lots of different motivational factors.
I think I wanted to echo the opportunity. So keep eye on all the opportunity. I think that's most important. So with my personal experience that I told you before, so I have very broad experiences. So moving from one space to from eye research to actually before I joined NISHD, I was at the National Institute of Alcohol Abuse and Alcoholism, which is totally different from what I think the part of the reason is that the opportunity. So you want to grab that opportunity and also be willing to learn new things. So, and I have never thought about I will move to an IAAA. To an IAAA, sorry, I keep forgetting the mic. And but with that mentality, I think learning is lifelong process. So I think I really enjoy learning new things. So all my before, the training education is all about eye or disease and the research. So working at the national NIAAA, I learned a lot about, alcohol drinking that will impact your health and the behavior, because of my training is, the postdoc training is human genetics. So from there, I apply my skill to learn that the behavioral genetics is so related to alcohol drinking. And I had the opportunity with NICHD where, I learned what the clinical trials is, and also learn a lot about the, medication use in pregnant women and children. So I think it's a fascinating space. I've learned a lot. It's a great deal. So opportunity really matters. Yeah.
And I just to kind of add another comment. I don't, whether it's linear, whether it's non-linear, I don't think you can go wrong. I feel like my trajectory is non-linear, more out of necessity. I'm also first generation American, didn't really have much guidance, didn't really know what was available to me. I knew what I kind of wanted to do. So I kind of just moved on from one opportunity. It was like, oh, I like this, or I didn't like this. But I think the biggest key thing here is not being fearful of seeing if it will work or not, right? And being open. I think I was trying to find a good word as they were talking, but I'm nosy. So I was sitting on conversations. I was like, I didn't think that this was a possibility. This sounds like really interesting research. How do I get into that? Maybe I can pursue that opportunity. So I think the big thing in your career development is not to be fearful of, pursuing things that you might be interested in. Maybe it works out and maybe it doesn't, and that's okay.
Hey there. So I know all of us trainees, we're getting a lot of training, but a lot of it is kind of hyper technical, and that we're learning to do things that we may or may not use in our everyday work, in our next step in our career. And so I was wondering if you guys had some skills that you really wish you had spent more time developing early in your careers, and if you could tell me more about those. Thanks.
I put my nickel in for statistics. That's always, you sit there working on a clinical trial, and you work with a statistician, and all of a sudden you realize we're not even talking English together. Like, oh no. So I think that, going back, I finally succeeded now that I get most of what they say to me, and I can understand it. And, but I think that having a really good, strong basis in statistics would have definitely helped more.
I agree. I think statistics for me was a big one. I was introduced to coding to really understand like just the raw data, and I wish I would have spent more time in that. I don't know if it would have helped my career now, but for me, understanding that I think would have helped me to maybe like translate it a little bit better and like more palatable, like understanding the raw data. So that's something I think I wish I could go back and have spent more time on. But
yeah, I'm going to say something different because you're learning a lot of facts and a lot of laboratory skills and other things. But I think what's really going to help you in your career is understanding relationships with people and dealing with colleagues. And so I can't believe I'm saying this, but because, I haven't, kind of delved into the social aspects of what we do, but looking back at my career, it's all about people. It's about the people that I've dealt with when I was younger. It's people I dealt with more from an administrative standpoint. I have a small group now that I am an administrator for. So it is really about the relationship with people. So if there are opportunities in your program to kind of work with someone who understands those aspects and helps you understand some of those things better, that's going to be a big boost for your career. Statistic would help too.
I was going to say, we had our big course external evaluation of the network, and each one of us in leadership had to present. So our statistician presented that we're moving out to something called estimands. And the review panel looked at me across the table, and they were like, I've just heard the statistician, our eyes are glazed over. What's estimands? And I felt like, oh, I got this. I can explain estimands to you. And everybody else in the room went. So, you get to the point where you can actually understand and explain, but I think it would have been really helpful to have gotten that stuff to my head earlier.
Yeah, I kind of agree with all the sentiments. And I had a very quantitative background, enzymology, nonlinear modeling, statistics. I think it's a great foundation. I think everybody should have some of that, and coding nowadays, I think is up there as well. Coupling that with the social skills, the ability to have relationships, communicate. I mean, communication is so key when it comes to anything, because even just recognizing when people are starting to talk and it's not going well, just because they're not speaking the same language. I mean, they're all speaking English, but it's not the same English.
You're not communicating.
And recognizing those things and having your own technical background to be able to evaluate when somebody else knows their stuff, right? Even if you don't understand it, you can sort of tell when somebody knows their stuff. So coupling those two things, I think, is very, very critical. So I just really learned the people skills a little bit more after grad school. So what else are on your mind, trainees? What are you worried about? What can we help you with? Or is there anything you just want to tell us that, as leaders in the field, we should be thinking about?
Yes.
So I think I'm just curious, Zhao, you mentioned that your background is majorly the eye research, right? So what do you, and then as Gil mentioned that there will be, many applicants for that kind of job. So what unique skills or experiences that you think you possess that set you apart in this position?
So, well, really, I think that the experience for all, my, whatever the past is that I think that first of all, I think you really wanted to decide where you, first of all, what kind of job you want to be. So working at NIH, I know we are not, especially extramural, is not a bench scientist. So not doing research. You wanted to make sure you really enjoy that. So that's first thing. So moving from wanting to change field. So that is, as I said, the opportunity, right? And also why I'm not moving outside of because of the family reasons. So then I decided, so I have a limited option to choose, but you want to make sure that you really enjoy, this kind of environment. Now you wanted to maximize all the opportunity options you would have. I think I could go out. I think that's the one thing I regret in my entire life is that I didn't pursue the medical practice because the family reason. So sometimes I have to sacrifice, to support the family. So that's the only thing I regret, not pursue being a physician practicing, but I enjoy what I'm doing, so, and I think that, so, you have to do, again, to make sure what do you really, what's your decision? So a decision is not, if yourself is fine, but you have a family, so it's joint decision. So there are a lot of other factors you have to think about, making a joint decision. But overall, so I think I enjoy, learning, moving one thing to, from one space to different space, from high to, now the pharmacology therapeutics in pregnant and children. So I've learned a lot. Now you mentioned PKPD, I'm learning that too. So I really enjoy really. So, at this stage of my career, still try to learning a lot of things, they're really, it's sort of like inspiring when you learning new things. So, yeah.
Like to build a little bit on what Zhao is saying and have you discuss kind of the work-life balance and, how family has influenced your career or the choices you've made.
I feel like for me, I put a lot of that off for my training. And if I could go back, maybe I would change it. I'm not disappointed in the path that I took, but it is important to me now. So I think with all the opportunities that I sought post fellowship, work-life balance had a priority for me. So I would really think about on your list of things when you're looking for jobs, like is money your top, is location your top, is work-life balance your top? For me, again, it was making sure that I could do what I like to do, still be able to move up in my field, right? Still be good at what I do, but also be able to have like the distinction between like, my work life and my home life. Because I don't think you should have to sacrifice. I know a lot of us do. But for me, I think I did enough sacrificing. So I got to the point where that was a really big thing for me. And there's a lot of positions, so don't feel like you can't move into, and I know a lot of times they, will say in academia, oh, it's tough. You can really make the position what you want it to be, right? As long as that's a priority for you going in, right? I think you have to, I think you have to voice your wants going into the position that you're looking for, but you also have to be realistic, right? Like if you want to go into a position that's asking for 80 hours a week, 100 hours a week, you know, work-life balance might not be priority for that position, and you might have to step back. So I think NIH does a wonderful job of work-life balance. I have to admit, I'm super duper happy where I'm at. I get to do the things that I like to do. I get to help people. I get to stay, you know, adjacent to the science. I do miss the bench, but then I also get to have like my family life. So.
Yeah, FDA is very different too. Coming from, you know, both a clinical and an academic institutions before that. And so I was very impressed when I went to FDA with how much emphasis and how much effort they put into making sure that staff has a good work-life balance. And so it's actually a great place to work. It's different, very different than academia. It's different than some of the clinical rigors of being in a clinical environment. So it's really just, they really work hard at making it a nice place to work.
I think academics is difficult, but not unmanageable. And first, I want to remind everyone that that idea of superwoman doesn't exist. She's not real. We don't play one on television either. So that means that the idea that you're going to do everything correctly, 100% for all spheres of the things you have to do all the time is wildly unrealistic. That means if you need help at home with, you know, doing laundry or cleaning floors, then that's okay. You can get that. There's no reason that you have to do that and all the 600 other things that you have to do. It's okay to ask someone else to help, give that someone else the job, hire someone to do that. And I think that's really important, recognizing that that expectation that people have of you shouldn't be your expectation of yourself. I think also your work-life balance changes with the different ages of your children. I remember when my kids were young and we had to do carpool, and you know, I once let my husband drive the children to school for carpool, and he took our child and not the rest of the carpool children. He didn't get to do carpool again after that. But you balance it. So that meant that the other people who were in the carpool did the Monday through Friday stuff, and I did every weekend party between my husband and I carpooling. That was our contribution to the carpool. So you do different things at the different ages. And I was fortunate that as part of working for the network, I did a fair amount of traveling to our international sites, etc. I took a child with me every time I went. And I would tell the school they're coming with me for a week, give them their homework, I'll make sure they do it, but they're going to learn more in Thailand or in Cape Town or in Brazil than they would sitting in the classroom for a week. And that was a wonderful experience for the kids. It was great for me. I coordinated with the people who are visiting because often they had kids the same age. I said, all right, can my kid go to your kid's school for the day? Or can you pull your kid out of school for the day and let's have the two kids hang together and do stuff? And our children, you know, grew up meeting children from around the world. And when we have meetings in Washington, they would bring their children in and I'd bring my kids, and the kids would go to museums and sometimes they just sat and talked. So you do different things with your family at the different ages, and sometimes it works, sometimes it doesn't work, but you've got a whole circle of friends and we are part of a community. Some of them are physicians, a lot who are not. And you work with your community of friends to make sure that stuff happens or stuff can happen. And you're doing some things on some days, someone's doing some things on other days. You're not expected to do everything on every day.
Comments online from Ripsable. One is you can also be a clinician investigator with a family. It may not be on the same timeline as you initially expected, but it is possible. And then she also asks how valuable or not valuable is leadership training for career growth for someone who is not necessarily looking to become a department chair, but just wants to be a principal investigator and doing research.
I'm still waiting to get the training. I don't really know.
My comment is I wish I had more. I'm always looking for
I
it.
think that the training programs, some of them are quite valuable, but again, it's what you put in and what you get out. So if you're going expecting to have someone tell you everything and you're going to come and know it all, you're not. I think it's prepping for the position or the training program, talking to people who are there, getting some ideas about the mentors that you need, and practicing some of the stuff when you get back. All important parts. There's no one size fits all. And, you know, when I was preparing for taking over the network, the first thing I did is I talked to the Columbia School of Business people. And I said, I need mentorship and training from the business world, because running a network is not just about the science and not just about leadership. It's about the fiscal stuff and the business stuff. And I actually was fortunate to have a half-year tutorial from Columbia professors as to what I should read, what I should learn, who to talk to. And it was stuff that I didn't know.
Okay, so basically I wanted to go back to something that you had mentioned about like students reaching out to you to work possibly collaboratively with you. I come from an MD background. I'm currently 8 months into my postdoc fellowship. And I remember at the time, the first day on the job, my PI told me that, oh, if you wanted to do some work of your own, you're more than welcome to. Being from a background in med school, the first 2 years are very book heavy. The last 2 years are very clinical heavy. So you never actually get the chance to properly dedicate significant time into a field, which leads to kind of not knowing what you don't know. You don't really know where the gap is. And to going back to what you said, what is some advice that you would give perhaps to someone who would be in my shoes at the time to kind of know what the field is lacking, kind of know what the interest is currently?
So you don't really know what the field is lacking. It's like asking about the absence of something. You can't measure absence. I think it's really keeping up with reading and listening at meetings to what people are talking about, because then you get a sense of, oh, this is what they know. And then you sit there and think about, well, what didn't they talk about? What are they not saying? And sometimes asking those questions, you know, we talk about pharmacokinetics and pharmacogenomics all the time. And sometimes it's like, so I don't understand, explain to me this. And everyone goes, oh, that's an interesting question. We don't know that either. So I think it's taking upon yourself to read about something that's fun and interesting for you. If it's boring for you, you're not going to read it. But if it's interesting for you and you sit there going, oh, I need to think about that. That's the fun stuff. And then asking people questions about that, but from an educated standpoint. Nobody wants to be, you know, Dr. Google. I hate when people call me up, what's the dose of INH? Well, you could look that up. Don't ask me that, especially at 2 o'clock in the morning on Friday night. But looking and reading and then asking yourself questions, trying to find those questions out. And when you can't, that's the time to say, maybe I will look into that. All right, what do you others think?
I'd say one of the, I think, best ways to sort of identify gaps in where fields need to go is to listen to the Q&A at, you know, scientific conferences and meetings. I actually find sometimes, when I was much younger, I used to be like, oh, listen to the talk. I didn't really care as much about the Q&A. Now I'm almost more interested in listening to the Q&A, because the debate that goes on there, and when you have really good questions and challenge, you know, appropriately challenging, you know, what's being said or talked about, you know, there's a lot of stuff in there. So I would definitely really recommend listening to the questions at state conferences. Anyone else? Was there another?
Hi, I had a question. So for trainees like us, we might be interested in something right now, but we haven't had experience in that particular field. So for pharma, I would say that there are 4 major areas like academia, industry, NIH, FDA. So once you get into something, how easy is it to switch to a different area within these 4? Like how easy is it to switch between areas at different stages of a career? do you think it's easy and then someone can just go experiment with a field, or it takes a while, like, you know, you need to?
It's your question, I stayed.
I mean, I think that I can't. That's a great question. I feel like I'm a rookie. I don't know if I, you know, have that much wisdom to answer that question.
But it's hit you.
Yeah.
You had that question hitting you.
Yeah, it's just, I think there's going to be challenges moving, you know, I won't say like you're going to have challenges with one more than the other. You're going to have challenges. And I think each challenge is going to be specific to what you're trying to transition to. Like for me, if I wanted to move from the government back in academia, you know, I don't have that academic record, right? So I pretty much would be starting from the ground up. So I think the challenge in that is just putting in the time to develop that academic record. Is it impossible? No. Would you have to put in a different type of time and dedication to it? Absolutely. You know, if I wanted to move into pharma, I think my focus would have to be different, and I would have to kind of rewire my brain because, you know, what we do for government is completely different focus than for pharma. So I don't want to say it's difficult. I mean, I don't want to say it's impossible. Will it be difficult? Yes. Will it be challenging? Yes. But I think though that what's make it interesting, right? I like to be challenged a little bit. So that's why sometimes I kind of look into other things, like what kind of, you know, sort of like stimulates you and you know, makes you want to grow. So, you know, that change might be absolutely worth it. I don't think it's impossible. You just may have to navigate a little bit differently.
Industry and FDA have a very established pattern. In one case, it's people getting experience in the FDA and then being able to go to industry and double or triple their salary. So you spend 5 years at the FDA, get experience, and then you're very valuable to industry. But it actually happens the other way too, is that people get experience in industry, and then for whatever reason, they get to a stopping point, they can't move up, and then actually we hire them in the FDA because of their drug development experience. Now, it's less common going from academia into FDA. You don't see that as often, but it does occur.
Yeah, and I think another aspect about making those transitions is sort of the additional skill, the non-technical skills that you learn. Some of them are very technical, but leadership skills, working with people. So, you know, at some level, you know, moving between different positions and areas becomes less about your core technical expertise and more about your ability to lead people, to manage, especially if you go into supervisory roles. So that's another thing to keep in mind in terms of, you know, moving between different sectors.
My question is specifically. So what are some of the challenges you have on the face establishing your research program,
Research team, and leading your research team in academia?
So when I started at Stony Brook, I was hired right out of my postdoc as the only pediatric ID doctor for the entire county, population 1.5 million. And when I was hired, the chair at that time said, just go ahead and build a program. And I went, okay, great. I'm going to do that. And obviously I was stupid because that was an impossible task. And I decided I would get grants at the same time while I was building a program. So 2 years after I was there, submitted my first grant and it got approved. And I went, I don't know why people complain. You just write a grant, they send you money. How funny was that? So I think the answer is there are multiple steps, and a lot of it has to do with your personality. And a lot of it has to do with you're working hard and working effectively. You know, we all work hard, but sometimes we don't work effectively. We kind of, you know, run around in circles and don't get stuff done. So I think some of it is being very organized and also asking yourself, is the juice worth the squeeze? And I have a colleague from NICHD in Rohan Hazra, and every time we'd get on a call and I'd say, you know, I want to do this, he'd go, is the juice worth the squeeze? Is it worth it for me to give you money to do that by the time you get the answer? So you ask yourself that question very often, in academics. What am I doing? When I get the result of what I'm thinking about, is it going to be worth the time and effort that I put in? And after that first grant, I recognized I obviously cannot do everything, and I had a newborn at the same time. So that got a little complicated. But I realized that I had to hire people, and I had to go to the administration at the institution and say, this is what I'm doing. If you believe that pediatric ID has a place here in the county, then you need to help support it. And all you need to do is give me one person, get me started, and I'll build from there. And sometimes that's all it really takes, is one person who shares your vision, who's going to work as hard as you did. And the individual that they hired for me was my administrator, who 30 years later, and both of us now have 3 children, we're all grown up and married, we're still working together. And we're still doing stuff together. And that relationship and bringing other people in who share your vision, sometimes they fit, sometimes they don't. But within our division now, we have 25 people who share our vision, who understand that sometimes, like during COVID, we're all going to work crazy doing stuff that we never did before. I made everyone be IRB certified, city certified, so that we had clinical trials. Everyone could obtain consents, including our nutritionist and our social worker, because we have to get kids and adults on COVID studies. And it's part of it is leadership. Part of it is, doing the job that you're asking other people to do. Part of it is having a vision, and also recognizing when you make a misstep, don't do it a second time. I have a sign in my office, I make every mistake just once. And having people around you that tell you, I think you're going in the wrong way. Please reconsider, or I don't think you're doing this right, and listening to them. So I don't think there's one right answer. I think part of it starts with you. Part of it starts with getting people who are listening and thinking like you. And part of it is listening to them when they tell you you're doing something wrong.
All right, we've got 12 minutes left. Any more questions? Oh.
Thank you for all this discussion. I have a question to Gil. A simple question. Who are more smart, the people who are at FDA or the people who are working for the industry?
Look here.
I don't think I should answer that. Of course, the people at FDA think they're smarter, and people in industry think they're smart. No, I think at the FDA, there's a real appreciation for industry and what they brought us. And so I think if you want to look at collaboration between them, you only have to look back at 2004 and 2006 and the critical path that was established when drug development was basically going downhill fast. And so, and the FDA has always acutely understood their commitment to public health and the importance that industry brings to that. And so even in drug development programs, I mean, our approach is not, we can't do that. The approach is really to try to facilitate the drug development program. Make sure it's safe, of course, make sure it's effective, but to facilitate it because that's what contributes to public health. And so I think when you stand back from the outside, it looks like there really are, two opposing forces. That's not true. Actually, the two are working in concert to a large extent for public health.
So I just wanted to talk to our young investigators here to tell them a story about how you, as a young investigator, can make a difference. Don't think it's us seniors that are doing the difference, young ones too. So I was a young investigator at Stony Brook. It was in the early 1990s. I had only been there for a few years. And I like to go to meetings and write down stuff about new drugs. You got a code number and you looked who wrote it. And we took notes then. We didn't have Zoom and camera. Okay. So I'd go back to my office and kind of read my notes and keep them in my tickle file of, oh, this tickled my brain. It was so interesting. We had a very young baby who was born at 28, 29 weeks, had necrotizing enterocolitis and multiple ostomies. She was now 9 months old, and she got a VRE infection, vancomycin-resistant enterococcal infection. And as you know, if it's vancomycin resistant, there's nothing you can use. And she was septic on a respirator. The father was a doc, the mom was a teacher. I was the peds ID doc by myself there. And I said, wait, I read about a drug. I don't remember the name because it only had call numbers, but I remember seeing that poster and it worked against VRE in the lab. Why don't I check it out? I'll call them. The parents were like, go ahead. And I was very young and stupid. And I said, of course I can do this. I went to my file, I found the paper. I called the pharma company and I said, so do you have any data on children? And they were like, who the hell are you? Why are you calling us? So I said to them, I'm a clinical investigator. I think this is important. I have a baby with VRE and we have no drug. And I know that you have a drug and here's the call number. And they said, we're in the middle of our early post phase 1, phase 2 in adults, and we're not interested in marketing that drug for children because, children don't get VRE infections, only adults do. And I said, well, obviously you're wrong. I have a kid here with this. So they were like, I said, can you send me the drug? After they got off laughing on me, they said, well, if you write a clinical trial and you get your IRB to prove compassionate use and you call the FDA and they agree, we'll send you drug. So I said, of course I can do that. So I sat down and I kind of scrapped together a compassionate use study protocol. I sent it to my IRB for compassionate use. The pharmacy company and I got the FDA on the phone. The FDA went, the parents sign it and it's compassionate use. Okay. It was a little loosey-goosey then. Sure, you can get the drug. So they shipped me the drug in little brown vials. I waited for the FedEx guy on a Saturday to come to my office and give it to me. He gave it to me. We had to go to the pharmacy and make it up. And we infused it into the kid. And the kid did incredibly well. She, within 24 hours, was better, off the ventilator. And, I wrote it like it was a lab experiment, like I had learned in Rockefeller. Every hour, what did I do? I took fluid from the ostomy. I did bloods. I tracked everything. I wrote a book on it. And, I sent it back to the company and they were like, okay, you convinced us. We're going to write clinical trials for children for this drug, we'd love to have you included in it. I said, great, I have nothing else to do with my time. That's fine. And long story short, that drug was something called linezolid, Zyvox. And because of that discussion with this young, stupid, naive investigator, they said, okay, we'll write the trials. And, I did work with them. I did not, no conflict of interest. I didn't get any money for it. It was my free time. That company was then bought by Pfizer. And Pfizer was very into how they're going to market it and stuff. And they went back and looked at all their papers and they saw this trail of paperwork from me. I did this and what I did this and what I did this. So they called me and they're like, did you work for this pharma company? And I'm like, no, I'm in academics. And they were like, wow, this is an amazing story. So then we had to go to Congress and talk about the family came, the kid came, I came. We talked about drugs and children. Long story short, Pfizer, of course, moved very far ahead and did lots more stuff with linezolid, Zyvox, and licensing and all the other stuff, moved it into the central nervous system and stuff like that. And the Pharma Association actually gave me an award for doing that because they would never have considered moving that drug into children, certainly not into a young child that age group. So the answer is, you can do very interesting things as a junior. Don't be afraid to think outside the box. And don't be afraid to call people and ask them questions. People will talk to you if you talk to them with a reasonable question. And sometimes you do stuff out of your comfort zone, and that's okay. You can do that. And sometimes you get a surprise and it works.
Yeah, so we are wrapping up. That was very inspirational. And does any panelists have any other parting words or anything for the training?
Sure, how can I say anything after that?
That was great. It was great. Yeah. I'm telling you, it was, it was so funny to get the award from the Pharma Association of the US. I had to go and get an award for them, and like, you're giving me an award? That doesn't make any sense. They're like, yeah, well, look what you did. I'm like, all right, I'll try to do it again.
All right. Any other questions, or should we wrap it on that very high note? I don't see anything online. How about some applause?
Thank you. Thank you to Aaron and all of our panelists, and great advice and some wonderful stories to take with us and hopefully shape some careers. That concludes our virtual portion for today, and our in-person group here has the option to visit some of the labs here at UCSD as part of the MPRINT hub. For those of you online, thank you for your attendance, and we look forward to seeing you tomorrow about 8 a.m. Eastern time. I think we'll start about at 8:15 tomorrow. So thank you very much for joining us today. So for those of you here, I think Sydney's in charge now. If you are interested, she can give you some information. For those who are visiting, I think you have some ideas for supper around the hotel. Any thoughts on when you might be back that others might join?
Did that answer the question?
Yeah, so if people are interested in lab tours, they're going to be pretty brief. We can just meet out there in about 10 minutes, so you guys have time to get your stuff.
Very casually.
Yes, very casual. Then we'll be doing a San Diego tradition, which is going to get tacos. So if you guys want to do that, feel free to come with us. The labs are on the way to the tacos, so it's probably easiest to do the lab tours and then go to tacos. But if you don't want to do that, come find me and I will let you know where to go.